Good morning, everybody. It's a great pleasure to be able to start the third day of the conference. It's been really enjoyable so far, lots of amazing discussions, great new knowledge being put in. So it's my great pleasure to be able to introduce our first speaker of the day, Stephanie Seneff from MIT, if I'm not mistaken, and she's going to tell us about the exclusion zone in the context of surfaces of proteins, or molecules, if I'm not mistaken. So please give her a warm welcome.
Good morning, everybody. You can hear me. Thank you all for coming so early in the morning to hear my talk. As he said, sulfate in the glycocalyx is what I'm talking about. I first want to mention that the second person talking today is Greg Nye, and he and I have been collaborating on so far 3 papers together. It's been a wonderful experience. He's a clinician. He sees sick people and figures out how to fix them nutritionally. And this helps me to figure out the biology of how everything's working or not working in the body, depending upon toxic exposures. About 7 or 8 years ago, I learned about, you probably don't know what this molecule is over here. Does anybody know? You can probably guess if you know me. Glyphosate. And maybe you don't know what glyphosate is. That's the active ingredient in the pervasive herbicide Roundup. And Roundup is all over our food supply, especially in the United States. And it is causing tremendous issues in health. I think it's the reason why we have a healthcare crisis in America. It's a major player in the autism epidemic, the epidemic in diabetes and obesity, Alzheimer's disease, and many other things, various problems with the gut. I've devoted my life to glyphosate over the past several years, and I think I've got Greg Nye in with me on this project now. I've had a wonderful time collaborating with many different people, publishing many papers on glyphosate, explaining its really unique mechanism of toxicity. And I won't get into that too much here today. I'm going to focus mostly on sulfate, but I am going to talk about how sulfate homeostasis in the body is being disrupted by glyphosate which is wrecking the body's electrical system, which then leads to all of these different diseases that are compensatory mechanisms to try to keep the body alive, keep the blood circulating by sacrificing various organs. And Greg will probably have more to say about that as well. So our talks today are talked about also in our paper, which is going to be showing up shortly in the Water Journal. And so look for that. If you want to learn more about what we have to say, you can read that paper. Okay, so the broad picture, water plays a vital role in human physiology, but it needs sulfate to work properly. And you all know about structured water, and thanks to Jerry Pollack, he really introduced me to the whole concept of structured water. And I've used his work tremendously to motivate our theoretical developments. So structured water maintained by sulfate supplies electricity to the cells. Cholesterol sulfate, which is produced upon sun exposure, provides sulfate to the vasculature. And toxic chemicals, especially glyphosate, disrupt sulfate synthesis and sulfate transport. And this induces an inflammatory response, which leads to systemic cellular injury. And then my conclusion is that sulfate deficiency is behind most modern diseases. So here's just some pictures of the glycocalyx, the artery cross-section over here, and you can see all these little hairs. That is the glycocalyx. It surrounds all of the arteries, all the veins, all the capillaries. A blow-up over here shows you all these tangled things in here. This is going to be very, very important. These are the sulfated glycosaminoglycans. We'll say more about that in a moment. And they maintain the structure. Question? Slow down. Oh, foreigners, I see. Slow down. I have a problem with speaking too fast. I'm so sorry. My brain works too fast. So this is lining the blood vessel, all of these complex sugar chains that are sulfated. And I'll have more to say about that in a moment. So I think that blood circulation is a central problem in the body. The body's always working towards making sure the blood can circulate. 2/3 of our water, of our mass in our body is water, but that represents 99% of our molecules. So we're basically, in some sense, a bucket of water with a few extra things thrown in. Most of the water in the body is gelled, and you learned about some of that yesterday. Gelled water in the cytoplasm, mostly gelled. But the blood has to flow, so it can't be gelled. And this presents a problem to the body because sulfate gels water. So you can't just throw a lot of free sulfate into the blood. You have to transport it using special molecules. And that, if that transport system is broken, you're going to have a systemic sulfate deficiency problem. And so blood delivers nutrients and removes waste from the cells, from the tissues. And there's gelled water that lines all the blood vessels in the body. And if you think of Jell-O, if you think of sort of putting Jell-O all along the edge of the blood vessel, the capillary, you present a very slick surface for the red blood cells to be able to slide almost frictionlessly through the capillary. Otherwise, you have all these things sticking in there that are going to cause friction and slow things down. So that's an important reason for the gel, to make a very slick surface for the red blood cell to slide through the capillary. So it also keeps the water gelled. It protects the sugar that's in the glycocalyx from oxidizing and causing, from glycation damage. You can damage the hemoglobin and all the blood proteins if you have a lot of free sugar in the blood. But there's a way to store the sugar safely by hiding it inside the glycocalyx behind the easy water, because it excludes solute, so the sugar becomes unable to react with things. Okay, so sulfate is crucial to maintain the structured water. And so the cells in the body all have this extracellular matrix, not just the endothelial wall, which is what lines the blood vessels, but also all the other cells in the body have an extracellular matrix that's formed from these complex sugar chains with the sulfates attached to them. And the sulfate ions bind to the sugars in a strategic, non-random pattern that varies over the entire body. So when sulfate is deficient, the pattern changes, and when the pattern changes, the behavior of the cell changes. And there's a very complicated communication network for the cells that involves these sulfated sugar chains, where different molecules bind to them and signal to the cell to behave in different ways. So it's a very, very important component of the cell. a place to store sugar, a place to make sure that the exterior is exclusion zone water, and then a way to be able to connect to the world through these other molecules that come in, signaling molecules that come in and give messages to the cells that allow them to behave in a way that's appropriate given their environment. So here's just some pictures of these of these various sulfated glycosaminoglycans. We can call them GAGs for short. As I said, they're in these extracellular matrix. Over here we have various specific molecules. At the top is chondroitin sulfate, and then keratin sulfate, and then heparin sulfate, and then heparin. Heparin is the most highly sulfated molecule in biology. And heparin plays a very important role in many biological processes. So you can see all these boxes around these sulfates that are attached to these sugars, which is keeping the sugars safe so they won't react with blood proteins and blood fats and cause glycation damage. And so here's then a schematic over here of the capillary where you have this glycocalyx right in here, the endothelial cells lining the blood vessels. They're producing this glycocalyx. These are red blood cells circulating through the capillary. This is an expanded view of this piece right in here. And these little jagged boxes are the heparin sulfate chains schematically. And then you have these things like this EC-SOD and this AT3. These are things that bind to the heparin sulfate. They have specific amino acid sequences that allow them to bind to the heparin sulfate. They get attached there, and then the whole thing gets broken down repeatedly, broken down and then built again, broken down and built again. The heparin sulfate only survives about a couple of hours in the glycocalyx, so it's constantly made and broken, but in the meantime, it's taken up into the cell along with all these things that it catches, and they then convey signals to the cell that causes changes in cell behavior. It's an amazing system. So this is the GAG chains. I've said all of this, and then this is the gel layer, the exclusion zone water, which protects, which hides the sugar so that it doesn't react. And then these signaling molecules, so the proteins like the antioxidant EC-SOD and the clot protective AT3. Those are those two that I pointed out. They hook onto the heparin sulfate with specific amino acid sequences that allow them to attach. Now this is a picture from Gerald Pollack's work, and I've basically mapped it to the endothelial situation. And so you have these beautiful hexamers of water in the structured water, and those are being formed in the glycocalyx in my case. So I'm mapping from his material where he has these Nafion material that he uses to create a simulated biological surface. In the real world, you have the endothelial cells that are creating this glycocalyx, which then creates this easy water zone that becomes negatively charged. And it pushes the protons out so that you accumulate protons at the surface. They can't get in because of the structure, but it creates a battery. And that becomes the energy that drives the electrical system of the body, I believe. And so these protons then, this is a quote from his work: "The surface energy of water is a non-metabolic fuel source for the molecular motors of our bodies." And I like that quote. I think that's true. And so now we will go on to our next section, electrical supply and blood flow. Here's a picture of the blood vessels. You know, it's sort of an electrical supply system for the body. Inside the cells, you have the cytoskeleton, and I suspect that is also a component of the electrical system, that the protons travel along the cytoskeletal wires to deliver the protons to the critical intracellular units that need to become acidic, that need to have a high concentration of proteins. So here's a schematic, a very loose schematic of a cell. And I've got these pluses, which are the protons being gathered along the edges of the easy water. And then the protons come out into these units, these little caves called caveoli that form in the membrane of the cell. I believe that the protons are ushered into those caveoli, and then they close off, and they eventually mature into endosomes and lysosomes, shown here, with decreasing pH as they gather more protons that are being delivered along that wire. So this is how you can create a lysosome, which is a super important unit in the cell for digesting cellular debris and for digesting nutrients, to supply the nutrients to the cell. So the the protons are swept in. They provide the charge that creates the low pH of the lysosomes. And the same thing probably goes on with the mitochondria. Both the mitochondria and the lysosomes hook to the cytoskeletal structure in the cell. And I believe they're able to pull in those protons that are being supplied through the battery that's being created in the extracellular matrix. And so there's the structured water. Okay, so this is a schematic of a capillary cross-section. And here you have the red blood cell taking up most of the space inside the capillary. And you have sulfate. I've just shown these sulfates in the glycocalyx that are maintaining the structured water, the EZ water. And then you have these protons that are gathering here. And negative charge on the red blood cell is due to cholesterol sulfate, in part to cholesterol sulfate that the red blood cell is continuously making and then delivering to the tissues. And we'll get into that part in a moment. And so I think we have maybe some things here. No, that just goes straight in. So yeah, the red blood cell can slide through in part because of the slick walls of the easy water zone, but also because of the charge, the negative charge repels the negative charge inside the endothelial wall. So it comes through almost hydroplaning through the capillaries with very little resistance. And now the other point to make is that the blood itself has a lower pH in the veins than it has in the arteries. So that's where I've indicated a minus over here and a plus over here. This is the vein side. This is a red blood cell inside a capillary. The red blood cell is negatively charged, in part because of the cholesterol sulfate. That negative charge, it becomes a moving negatively charged particle in an electric field, which creates a magnetic signal that the cells in the endothelial wall respond to. And so you have, this is a schematic of the magnetic field. The endothelial cells have been shown to release nitric oxide. Let's see. They release these these are nitric oxide down here. They release the nitric oxide in response to the signal here. And when the heart rate goes up, the red blood cells are being pushed in, creating an AC component that will also go up with the heart rate, and that will increase the amount of nitric oxide that's released in a very neat system that provides the nitric oxide is a signaling gas that relaxes the artery wall, and so the red blood cells can flow very smoothly in the response to the increased heartbeat. So endothelial nitric oxide synthase is an important enzyme that I'm going to talk about next. I call it a moonlighting enzyme, and I've written papers, more than one paper, describing my theories for the fact that E-NOS, it makes nitric oxide quite obviously by its name. It's an endothelial cell protein, and I believe it also makes sulfur dioxide. I think it has two different output products depending upon circumstances responding to signals. So here's just an interesting story about heart disease related to sunlight. Each of these dots is a country, and this paper, they estimated the amount of total sunlight exposure over the course of the year, considering the latitude and the amount of rain. And then they looked at the death rate from heart disease, and they found a pretty strong inverse correlation. Countries that have a lot of sun and that are low latitude have low rates of heart disease. So there's some relationship between sunlight and heart disease. I believe it's connected to the fact that we can use the sunlight for energy to make sulfate. So here's a provocative proposal. Cholesterol sulfate supplies sulfur, oxygen, cholesterol, energy, and negative charge to all the tissues. Sulfate is synthesized from sulfide in the skin and in the bloodstream, utilizing the energy in sunlight. It protects from UV damage and it keeps microbes out in the skin. And endothelial nitric oxide synthase is a critical enzyme in the process of making the sulfate. And so I would say that the skin then is a solar-powered battery because it can make the sulfate from the sunlight and hold the energy of the sun in the sulfate. So bold claim deficiencies in cholesterol and sulfate supplies to the blood and to the tissues are the most important factors behind modern diseases. So RBCs express E-NOS. It is a very interesting point. People realize, researchers found out that RBCs, red blood cells, make E-NOS. Red blood cells don't have a nucleus, so they have to decide in advance what proteins they're going to make, and then they're stuck with those proteins once they've lost their nucleus. They make E-NOS, and they surely don't use it to make nitric oxide, Because if they did, they'd be killing their hemoglobin. Nitric oxide is very much like carbon monoxide. It will bind to the hemoglobin and prevent it from carrying oxygen. So there's no way that red blood cells would want to be doing that, which motivates me to think they use their E-NOS for something else. So they actually work hard to make sure they don't make nitric oxide by not having any transport mechanism to import arginine. Arginine is the precursor to nitric oxide. The red blood cells don't actively take it up. If they do accidentally take it up passively, they have arginase that will break it down. So they're making sure not to have any substrate for their E-NOS protein. And then their E-NOS is also in the wrong place. It sits on the cell membrane instead of being inside the cytoplasm, which is where it needs to be if it's going to make nitric oxide. So then the question is, what do they use their E-NOS for? I believe they use it to make sulfur dioxide. And it's been shown in papers that they can extract sulfur from sources like garlic. Garlic has a lot of different sulfate and sulfur compounds. And red blood cells can take up the sulfur from garlic and release hydrogen sulfide gas, and then they can actually use their hemoglobin to oxidize the hydrogen sulfide into thiosulfate. The thiosulfate then, I believe, gets converted into sulfate using E-NOS in the red blood cells. And they use glutathione, I believe, to hold that sulfur momentarily as SSH, an extra sulfur in the glutathione. That particular sulfur atom that came from the garlic as hydrogen sulfide gas will eventually then get oxidized to sulfate using E-NOS. So here's a picture from a paper by Bianco et al., published in 2018. Very interesting paper because it shows that hemoglobin, here we have HB here is hemoglobin. It gets oxidized to hemoglobin 3, and then it picks up this sulfur from the garlic, and it can then convert it here to thiosulfate using the hemoglobin to do that. Very, very interesting, because then I think the thiosulfate becomes available to become sulfate using E-NOS. And so just schematically here, you have 2 hydrogen sulfide atoms. Hemoglobin is turning them into thiosulfate. E-NOS is then pulling off this extra sulfur on thiosulfate, oxidizing it to sulfur dioxide, and eventually it produces 2 sulfates using other oxidizing enzymes to produce sulfate from sulfur dioxide. And so here's my hypothesis. E-NOS is a moonlighting enzyme. It produces nitric oxide when it's phosphorylated, when it's in the cytoplasm. It produces sulfur dioxide when it's attached to the membrane and unphosphorylated. And the electromagnetic signals coming from the blood dictate which one it uses. And these two switch on and off back and forth to control the blood viscosity and to control the glycocalyx formation and destruction. And so this is how it goes. Then you have sulfate is a cosmotrope, so it gels the blood. Nitrate is a chaotrope, so it relaxes it. It turns the blood into water. The blood can then titrate between the two to get the right mix. But at the same time, the sulfate can build up the glycocalyx, and the nitrate will tear it down, because nitrite is a molecule that breaks down the glycocalyx. And so this works out very nicely to go back and forth between building up the glycocalyx and breaking it down. And so the problem is that E-NOS is very vulnerable. It's vulnerable to a lot of toxic chemicals in the environment. It depends on glutathioneal cobalamin. It has which will then mean cobalt, the mineral cobalt, and glutathione and vitamin B12. So these are all crucial problems if you have deficiencies in these things. It depends on heme, and it depends on heme iron, sulfur, zinc, and of course oxygen. It depends on NADPH, and it depends on sunlight. So there's all these different dependencies that can cause E-NOS not to work if they're not supplied adequately. It's also an orphan cytochrome P450 enzyme. And glyphosate has been shown in experiments to suppress cytochrome P450 enzymes. So that's going to mean that it will suppress E-NOS through that same mechanism. So glyphosate is a train wreck for E-NOS. Here's some of the reasons why. I mentioned the CYP enzyme. Glyphosate suppresses that. It depends on heme. Glyphosate has been shown to suppress heme synthesis. It depends on an essential terminal glycine residue to bind to the membrane. And my theoretical treatment of glyphosate in my previous papers that I've published with many colleagues, we're claiming that glyphosate substitutes for glycine during protein synthesis by mistake, and that is a critical part of glyphosate's insidious cumulative toxic mechanisms. So glyphosate can be substituting for glycine in E-NOS and messing up its ability to attach to the membrane. E-NOS also depends on zinc and on iron, and both of those are chelated by glyphosate. Glyphosate is a major mineral chelator. So these are all different ways in which glyphosate can be expected to disrupt E-NOS. So here's just a picture of a schematic of the E-NOS dimer. It forms this double E-NOS with a cavity here. There's a zinc atom in that cavity. Research papers don't know what that's for because it's not used to make nitric oxide. I believe it's used to grab the sulfur to oxidize it to sulfate. And that the zinc is critical for that to work. And then there's the glycines that are essential for hooking it to the membrane and also for forming the dimer. So if those glycines are substituted by glyphosate, both of those will break down. So E-NOS uncoupling, this is a paper that talks about uncoupling of E-NOS. There are many papers that talk about this, where E-NOS normally makes nitric oxide, but in its uncoupled state, it produces superoxide instead. And superoxide is, of course, very damaging. It's a very damaging oxidizing agent, particularly when the two of them combine. The nitric oxide and the superoxide combine to form peroxynitrite right here. Very, very nasty, very highly reactive oxidizing agent. So what I think is happening is that when E-NOS is mistakenly producing superoxide, it means to be producing sulfur dioxide, but it's broken. And so that's how you get in trouble. Normally it would go from superoxide to sulfate, from nitric oxide to nitrate, but it can't do this the sulfate path because of the disruptions by glyphosate. And so, again, this issue of building up the matrix and breaking down the matrix in a constant yin and yang. And so, again, it's regulated by the flowing blood. So this is called "oh no" in the vernacular because it's extremely toxic. So here's a nice paper if you want to learn about E-NOS uncoupling. And it talks about how angiotensin II, which is a stress response protein, it produces... It induces NOx, which is NADPH oxidase, which produces superoxide. And then NOx actually encourages E-NOS to make superoxide as well. So you get a profusion of superoxide production that really wrecks everything. And in the picture, the paper has this picture of the fire with the superoxide because E-NOS is doing that. And that's because of these G's here. These are glutathione binding to the cysteines in the E-NOS and messing up its ability to make nitric oxide, trying to get it to make sulfur dioxide, but not succeeding because of the disruptions by the toxic chemicals. And so my hypothesis is that E-NOS produces superoxide instead of sulfur dioxide when it is disrupted by glyphosate. This interferes with sulfate production and it induces oxidative stress, insufficient sulfate in the red blood cell membranes and in the vascular wall, impairs blood circulation and impairs the electrical supply. And this defect may be the reason for cellular damage due to the peroxynitrite in multiple diseases linked to inflammation. Okay, how to stay healthy. Eat a strictly certified organic diet. Get rid of the glyphosate that way. Get lots of dietary sulfur. Eat sulfur-containing foods. Cruciferous vegetables, onions, garlic. Garlic is my favorite. All kinds of seafood, fish, cheese, grass-fed beef. These are all good sources of sulfur. And get out in the sunlight. Don't use sunscreen. Don't wear sunglasses. I think it's important to get sulfite exposure to the pineal gland behind the eyes. So enjoy the sun and get grounded. Best thing to do is walk on the beach, in the water. You've got really good grounding, pouring in the negative charge, and helping to keep your electrical circuit working properly. So in summary, the human body utilizes sunlight to make sulfate, which maintains gelled water surrounding the cells and the tissues and supports blood flow. Gelled water induces charge separation that supplies electricity to the tissues. E-NOS is a moonlighting enzyme that switches between nitrogen and sulfur oxidation based on electrical signals coming from the flowing blood. The sulfate builds up the matrix, the nitrate breaks it down. E-NOS is highly vulnerable to poisoning from glyphosate and other toxic chemicals such as aluminum. When you put the aluminum in the sunscreen, you're messing up the E-NOS in your skin that's making the sulfate. Organic diet and lots of sun are the best paths to good health. And thank you very much for your time.
Thank you very much for this great talk. Always learning a lot. and I retain that the solution is to go to the beach, which is great.
Yes.
Question from Jerry. There's a mic here.
Thank you, Stephanie, as usual, for your provocative and interesting presentation. I'm wondering, your emphasis is on sulfate, but we found in our experiments that, for example, phosphate is also an ion that builds easy water, and Gilbert Ling talks a lot about that, especially ATP as being a nucleator of growth of structured water. Do you think that the phosphates also play such a role in the body?
I absolutely do, and in fact, I'm writing a book on glyphosate right now, also on sulfate and phosphate, so I go into a lot about both the phosphate and the sulfate. And phosphate in particular is very critically disrupted by glyphosate. Glyphosate disrupts proteins that bind phosphate, and this is going to mess up things having to do with ATP. So I think it's possible that the ATP and the phosphate inside the cells is important for maintaining the structured water inside the cells.
Thank you very much. It sets off a cascade of inspirational ideas that pop into my mind. And one is regarding the nitric oxide issue. There's a feedback on the speakers. Nitric oxide issue, particularly upsetting the balance between nitric oxide and the endothelial lining when you consider that bad cholesterol is attaching and maybe consuming, digesting, disrupting first the easy layer and then disrupting endothelial lining, therefore inducing arteriosclerosis. So maybe you can say something on that.
Arteriosclerosis, yeah. I have my own opinion about arteriosclerosis. I believe that the cholesterol that's stored in the artery wall supplying the heart is actually, it's like a squirrel storing nuts. It's actually storing the cholesterol, waiting to become cholesterol sulfate when sulfate supplies arrive. And so it's because of the sulfate deficiency problem that the body needs to make cholesterol very readily available to the heart so that when the sulfate's available, the cholesterol is actually released as cholesterol sulfate into the HDL particles. And then that cholesterol sulfate can be delivered to the heart in particular, but to the tissues in general. Because I think there's a systemic cholesterol sulfate deficiency problem in the body that is driving heart disease. Mm-hmm. Some resonance with that. That didn't quite answer your question about the nitric oxide, but the nitric oxide actually oxidizes to nitrite, and then the nitrite strips away the heparan sulfate. So if you keep eroding the heparan sulfate in the glycocalyx without replenishing it because you're stuck trying to make sulfate, you're going to thin it down. You're going to have a deficiency in the endothelial matrix, in the supply of sulfate in the matrix, which is going to then make also the blood very, macrophages can cross the blood barrier and get into the tissues and cause inflammatory responses in the tissues.
There is a whole story of modification of the glycocalyx in cancer cells. You know, in cancer cells.
Cancer cells.
Transformed cells
Yes.
have partly destroyed glycocalyx. This
yes.
can be shown, it was shown a long time ago by electron microscopy using the ruthenium red dye.
Oh, wow, yeah.
And this would explain at least the fact that cancer cells are also more resistant to grow in agar containing dextran sulfate. Probably the charge in ions is different in normal cells and cancer cells.
Yes, yes. And I think Greg Nye is going to be talking about cancer and sulfate in the next talk, so that will be very timely. Thank you.
Yeah. Thank you. This was extremely insightful, though very fast.
Sorry about that.
I was wondering, in the whole debate about glyphosate, people talk about rats getting cancer or not. But isn't there a more simple sulfate assay or nitroxide stress assay that could be applied, or can you get hold of the data on those tested animals that could be applied in every laboratory to check the level of damage before the cancer actually breaks out?
I think there's many, many studies that could be done on glyphosate related to my theoretical work. I don't have a lab. I would love to do those studies. I keep hoping that people such as this audience would be inspired to do the appropriate studies that would test my theories. I do see lots of evidence from the studies that have been done in terms of depletion of glutathione in the liver and oxidative stress. It tremendously produces oxidative stress. It damages DNA. It causes fatty liver disease. That's been shown even at very low levels. It disrupts the reproductive system. It attacks the germ cell. So there's many, many papers that have shown glyphosate causes harm. But people would be much more directed towards specific experiments linked to my work if they would decide that was something worth doing. And I really hope that people who have the facility to do that will do so.
Good morning. Thank you for a very provocative presentation. I've used glyphosate as a farmer, and I know a little bit about the science of glyphosate, and my understanding of some of the science is that when you spray it, and it comes in contact with the soil, the organisms in the soil break it down very, very quickly. So in fact, if you put a living plant on it in about 2 hours after you spray it, it has no effect on that plant. So it's not glyphosate anymore in the environment. What's your understanding?
That's actually not true. It's true under optimal conditions, but there are many conditions in which glyphosate survives in the soil long term. In fact, it's been shown in studies in Brazil that glyphosate is accumulating every year, that they keep using the glyphosate on these GMO crops. The next year, there's more glyphosate in the soil than there was the year before. They've shown that in studies over repeated years. People have found certain soil types where it can last over multiple years. It can still survive in the soil. So it depends on the actual microbes that are in the soil and also on the amount of organic matter, the amount of silica. Various soil types are able to retain the glyphosate for a long time. It also survives in the ocean. A study on glyphosate in the ocean showed that it was still around. A significant amount was still around after a year of the glyphosate exposure in the ocean. So it's not true. Monsanto likes to claim that it disappears very quickly, but that's not true. It's also been shown that it accumulates in the tissues, and that's been shown by Monsanto researchers back in the 1980s. They did unpublished studies where they fed animals radio-labeled glyphosate, and they showed that there was radiolabel in the tissues, especially in the bone marrow, which is a very dangerous place to accumulate glyphosate because it's going to affect the immune cells. It'll get into the immune cells and cause problems.
Okay, well, thank you very much for your very good presentation. It was very informative to me. In the Oriental medicine, it has been known that the sulfur is very important for the long life and health. Now, from the lecture today, I understand why it was so. And since the sulfur is the toxic materials, toxic elements, so people tried to
Right.
have a dog to eat the sulfur and some that kind of way. And also garlic is an important organic supplier of the sulfur. So just a comment for you that it has been known the importance of the sulfur from a long time ago.
That's good to know, yeah. I really feel sulfur is way underemphasized. We don't even have a minimum daily requirement for sulfur, so people don't really think about sulfur deficiency.
I think we have to move on. So let's thank our speaker again. And then
So now the next speaker. Sorry, I must help me this. Greg Nye. Yeah, this is it. And his speech will be about cancer, sulfate, toxins, and biological water. So, here we are.
Thank you. Hello? Maya? Yeah, okay, good. Okay, so, and I want to start also just with a nod to Stephanie, because I feel like much of what I'm doing in the clinic, not just me, but Maria, who I work with and is a nutrition therapist that I'm collaborating on all the patients with, in many ways, it's a large part of what we're doing in the clinic is translating a lot of the work that she's done around sulfur and its effects on the body and how that plays out clinically. And sulfur is absolutely essential for health, there's no question. The issue is that there is this growing number of people who are having problems metabolizing sulfur, and it causes an enormous range of symptoms, diseases and symptoms. And I'll talk a little bit about why people are having problems with sulfur metabolism. I'm going to focus today on cancer. Cancer is probably about 60 to 70% of our clinical work. Then we're dealing with a number of other, inflammatory bowel disease and all kinds of inflammatory diseases that I and Marie and I both believe are often related to sulfur problems. And when we deal with sulfur and sulfur metabolism, it is astounding how many of these conditions resolve. So I think that it's tragically unrecognized the role that sulfur is playing in many of these diseases, and that once that becomes better understood, then a whole new range of therapies will open up. So I'm going to talk mostly about cancer today with some reference to other things. Let's see, do I have to do this side manually? So just to get the scope of the problem and put it in context of iron, which we all kind of understand iron, there's a lot of awareness about the need for iron. We have about 3.5 grams of iron in the body. We have about 140 grams of sulfur. So sulfur, we have lots and lots of sulfur constantly needed by the body. A very large portion of it has to get moved over into production of those heparan sulfate proteoglycans that Stephanie was talking about and that I'll talk more about. The sulfur in the body is playing an enormous number of different roles, and many of these roles are because of this incredibly complex coding system that's happening within the glycocalyx and those heparan sulfates, and how they sulfate those proteoglycans are sending signals, and those signals are allowing for lots of different things to take place. Keeping in mind that sulfur in the body has only one source, really, and that's dietary. All of the sulfur has to come in some way through the diet. Now, we're not taking in sulfate, or very little amount of sulfate. We're taking in sulfur in various other forms, and our body has to go through processes that get it converted into sulfate, where it plays its role in these heparan sulfate compounds. There's another supply which is internal, so the mucus has sulfur embedded in it, and that sulfur can actually be recycled and turned into sulfate. But we all have to take in sulfur in those garlic and cruciferous and all of those sulfur-heavy foods. We have to go through these enzymatic processes that then get converted into sulfate. And so what happens if we can't get through those processes? Then things start to fall apart. So 2 big questions. What if the supply runs low, and why would it run low? So to start with the second question of what happens that would lead us to not be able to produce the sulfate in the way that we need to. So there are genetic issues, and I do reviews of the polymorphisms around sulfate production with lots and lots of people. Looking at these and other enzymes, CBS and CTH, and I won't go into all that, but suffice to say that when somebody has, for instance, a polymorphism in that sulfite oxidase, SUOX, if there's a polymorphism in that enzyme in particular, which I don't see very often, I've only seen it 4 or 5 times, that person's sulfur metabolism is always going to be a wreck. Like, it's very hard to work with somebody who has an issue there. So now we can look into genetics, and we get to see these things that we couldn't see, 20 years ago. There's a dietary issue. So lack of cofactors that are needed. Molybdenum is probably the one that's best known because it's a cofactor in that sulfite oxidase enzyme. But there are other cofactors. Stephanie just talked about glutathione and B12, other enzymes that play a role in this processing of sulfur compounds to get them to the end product that they need to be. And then, as Stephanie just talked about again, these heavy metals and environmental issues that also can clog the system up. Many people are dealing with all three of these things. And I think that a reason that sulfur problems are becoming more and more prominent is because sulfur is a primary detoxification atom. It's a chelator. If you take any detox product off the shelf of a health food store and you look what's in it, it's full of sulfur compounds because sulfur is involved in detoxification. The problem is that sulfur is also involved in structuring water out here in the periphery. It's not supposed to be detoxifying out here. It's not supposed to be binding out here. But now we're exposed to all kinds of things that we were not exposed to 100 years ago. And so when sulfur binds up systemically, we run into big problems. So what happens if we run low on sulfate and the supply that we need? Well, our bodies need to go through some compensation mechanisms. We got to go to backup plans. So a first thing that we can do is to extract some of that sulfur from the mucous membranes, acute illness, right? And I remember when I first, reading about the sulfur and how it's built into these, into the mucous membranes, and thinking, oh my gosh, maybe when we get a cold, what we're really trying to do is to resupply sulfur, and had that thought, and I send this note off to Stephanie saying I had this idea, what if, and of course she'd already written a paper about it or something. But acute illness is potentially a way to extract the sulfur needed in the short term, and there are gut bacteria that will take that, particularly that sulfur, and recycle it. Will generate hydrogen sulfide and sulfite, which can then become sulfate. Well, what if that doesn't meet the need? Well, there's another thing we can do, and that is to start overgrowing sulfur-fixing bacteria in our guts. And this is something that Marie and I are working with lots and lots in the clinic, because there's this whole phenomenon, SIBO, small intestinal bacteria overgrowth, which is very prominent now. This process where people are having just chronic digestive and bloating and just misery in their gut. And the typical approach to that is to kill the bugs. That's both the conventional and the naturopathic approach generally. What I think is that these bacteria are necessary. It's not going to work to kill these bugs because they're trying to supply something that our body needs. Because they will take dietary sulfur. They'll generate large amounts of hydrogen sulfide, which is a bummer because that causes symptoms. And those symptoms range from skin issues and rashes and headaches and hot flashes and brain fog and fatigue, and they all go with too high levels of hydrogen sulfide and sulfite. But it's filling a need that our body has to fill, and that is we have to have substrate that can make sulfate. Hydrogen sulfide is that substrate. So that is what I just call a pre-chronic. I just made that up, but it's just sort of like this, the next level beyond acute. And then, if that doesn't fill the need, we have to get into this chronic inflammatory state because in order to get from hydrogen sulfide to sulfate, you can get there through direct oxidation. You can in an inflammatory environment, that is what facilitates that conversion. And so, again, bummer, causes joint pain and, you know, all of the things that we associate with inflammation. But you can help to generate what the body is needing. And not only, I mean, it has to have this. Like, you can't maintain structure and function without adequate amounts. And then what I'm going to spend the rest of my time talking about is cancer, which I think is really, that's the end state of an extreme sulfate need. Now, of course, I'm not suggesting that all cancers are sulfate need, but I'm suggesting that one possibility for many cancers. And I'm not going to spend too much time on this, but I want to just make sure it's clear. The notion of cancer as a mutational disease, I think that I want to completely do away with that. Cancer is not a problem of mutations. Cancer is a tissue-based disease, and there are many, many studies that I think show this very well. Now, you can induce cancer, you can by mutating cells, but ultimately in people, cancer can be shown that it is really damage to the tissue that the cell is inhabiting that defines whether or not the cell is going to become malignant or healthy. And if you implant a healthy cell in tissue that has been damaged with chemicals or radiation, that healthy cell becomes malignant, regardless of the mutations in the cell, in the nucleus. And vice versa, if you take a fully mutated cell and you implant it in healthy tissue, that cell will become a healthy cell. The tissue is giving the signals that are telling the cell what to do. So, and again, you can do the same with the nucleus. You can take a mutated nucleus and implant it into the cytoplasm of a healthy cell where you've taken the other nucleus out. And even though it's fully mutated, it will go on to become it will divide into healthy cells, not into a malignant cell, suggesting that it's not the mutations in the nucleus that are determining these things. So I only want to talk about this to make sure that we're clear that cancer is a tissue-based phenomenon. The other point that I want to make is what I think is also accurate, and these are ideas by Carlos Sonnenschein and Ana Soto, who have written lots about this, that all cells want to divide. All of our cells want to divide. And we tend to think about cancer as being quiet cells that are then kind of attacked by growth factors that cause them to divide. But another way of thinking about this is that all cells want to divide, and that there are systems in place that inhibit cell division, that prevent the cell from dividing. And it's when the inhibitors fall away that the cells then can take off. And I think that there's very good evidence for that as well. And the reason I bring that up is because I think that this whole heparin sulfate system is very much part of the inhibition of cell division. It's what keeps things held into place, so that when that system messes up, then the inhibitors don't do their job. So what is the nature of the damage that is happening to the tissue that leads to cancer? Well, it's all the things that we know about. We do know that malignant tissue has less easy water. We'll call it easy water. I mean, this is what MRIs, MRIs are looking at, essentially the structuring of water that is happening within and around tumors, and finding that tumors have less bound water or structured water than non-malignant tissue. Jerry, in a recent article, showed that the glyphosate has shown that it will degrade the EZ, and we know that heavy metals degrade it, and exposures will degrade. All these things will degrade the EZ. So what happens if the body has lost access to adequate amounts of sulfate and thus is not able to maintain the structuring of the EZ water in the way that Stephanie had just talked about, this intimate role that sulfate is playing in ultimately providing that structure? Well, cancer is actually kind of ideal it's a perfect thing. And not perfect, but it is a way that the body can fulfill either a local or systemic need. And just as an aside, I think essentially all diseases are in some way adaptive, and I don't think there's any reason to think that cancer is otherwise. We know that there are lots of stage 1 and even stage 2 cancers that arise, and then they do their cancer thing, and then they go away. And Mina Bissell, who is in one of these references here, has documented this extensively, that many, many cancers cancerous tissue arises in us, it hangs out, and it leaves, and it never does anything. And she thinks we need a whole new labeling system for these cancers. We shouldn't even be calling them cancer because they were never going to do anything. So the question is, well, why are they showing up in the first place? Well, they're accomplishing something, I think, that is quite specific. These cancers are factories that generate high levels of these sulfur compounds, of GAGs and heparan sulfate proteoglycans. So inside the cancer cell, they generate these things, high levels. They ship it out. Outside of the cancer cell, there are very high levels, much higher than around normal cells, of these enzymes, heparinase enzymes, that will break down these compounds and essentially provide the raw material to the local, either the local tissue or perhaps systemically, if that's needed. They provide heparan sulfate or hydrogen sulfide for further production of sulfate compounds that might be needed. And so these tumors are the factories that can supply that need. And let's say you supply the need, you just shut down the factory. You don't need it anymore. It's a self-resolving tumor. It shows up, it does its thing, it goes away once the local need is met. Some other interesting upregulations in a cancer cell, 16 genes related to the production and export and processing of these sulfur compounds are upregulated within tumor cells. So these aren't random mutations. These are very specific. They're moving the cell in a very particular direction to accomplish something. And also the PAPS enzyme, which is needed to get organic or inorganic sulfate converted into the active form, or the organic form of sulfate. So the point being that these tumor cells are well designed to be manufacturing these sulfur compounds that the body may in fact be running in short supply on. You fulfill the need and then you can shut it down. When you inhibit sulfate transport, you grow tumors at a dramatically faster rate than if you allow adequate amounts of sulfate to be supplied, which is another argument in favor of the notion that a reduced supply of sulfate to tissue or cells means that you got to get that factory growing. You have to get that factory big enough to supply the need that is not being met elsewhere. And also, what is important to understand is that this process is coordinated. It's not like a random thing and the cell is moving in aberrant directions. These enzymes, the heparinase, there are two main forms of it that are coming into play here, heparinase 1 and 3, they are coordinating the amount of growth or restraint that is going on within the cell. So the point is that it's a coordinated activity. It's an intelligent activity. It's meeting a specific need. And what I would suggest is that if the need is met, then the tumor can go away. If you supply hydrogen sulfide exogenously within animal models of cancer, you can induce apoptosis. They affect cell proliferation. You restore the EZ. supplying more sulfate can have beneficial effects on cancers. In other words, if we go with this idea that a tumor is trying to supply sulfate, well, if we supply sulfate from the outside, it actually helps to bring some of that malignant process under control, which makes sense because now you don't need the factory to do it, it's coming in from the outside. And keeping in mind that the whole point of having this factory is to supply the sulfur compounds that help to structure the water, which is necessary to maintain health. And actually, the point that I didn't make here is that what happens if you've got your stage 1 or stage 2 tumor trying to supply the sulfate that's needed, either locally or systemically. Well, what happens if it doesn't meet the need? Well, you got to grow the factory a little bit. Or maybe you got to grow it a lot, and maybe you got to set up shop over there too, because you have to increase the supply that's not being met, which it wouldn't be met in the context of ongoing toxic exposure, for example, or heavy metal exposures or nutrition deficiencies or whatever. If the need for that factory grows, then you got to grow the factory. So in that context, then in my, thinking clinically, well, why would these compounds that, of course, come into many cancer treatment programs, and it's widely understood, at least in my world, the naturopathic world, that these are commonly used within the context of cancer therapy. Well, how do we make sense of that if this idea that tumors are sulfate factories, if that's true? Well, when Stephanie was just talking about glutathione and B12 as the role that they play in helping to maintain these heparan sulfate proteoglycans, CoQ10 is an essential cofactor in fact, I have a little diagram over there. It's probably too small to see, but of the heparan sulfate that is generated in the body, a large portion of it is enzymatically converted first to SO3 sulfite and then SO4 in order to get from heparan sulfide. In order to get from heparan sulfide to SO3, you have to have CoQ10. In 2 different steps, you have to have CoQ10 to make that conversion. And so a deficiency of CoQ10 would lead to a backlog. Not only does that deplete the body of SO4 ultimately, but it leads to a buildup of hydrogen sulfide, which causes the symptoms that we are seeing in the clinic all the time. Excess levels of hydrogen sulfide, brain fog, and really, it's dramatic. When you get sulfur metabolism working, ultimately what's happening is hydrogen sulfide levels are dropping and symptoms get relieved in sometimes really dramatic ways. So, all these taurine, all of these have their own role to play in cancer therapy. And I have just picked a couple of them out because these talks are short, but you can find very clear links between the roles of many cancer therapies, alternate natural cancer therapies, and the impact that they have on allowing the body to adequately process sulfate. And when you compromise glutathione, you increase cancer risk. If you deplete B12, you increase cancer risk. I think it's interesting to note that DMSO also has a role in maintaining water structure. DMSO is something we use very high concentrations as an IV therapy, has really impressive activity in terms of anti-cancer activities. The smell can be oppressive sometimes, but because people smell like oysters when you put a bunch of DMSO in an IV, but there are really some great qualities to DMSO as a differentiating agent, And also, because of the structure, it's thought that it actually looks like water to the body and maybe is utilized in the same way. But so DMSO is another one of these cancer therapies that I think is, can be quite important. Let's see, why aren't sulfur-based? Oh yeah, why? So then, the thing that keeps me awake is why, if this is true, why is it not true that when you just do sulfur-based therapies, like the kinds of diet program that Maria has, kind of generated and patients are going through, she's really doing the hard work. Actually, I just get to prescribe supplements, but she has to get people to actually change how they eat. That is not a game that you want to be in with a lot of people. But that is, that's where the gold is. That is the key to this whole thing. And it is confusing to many because, well, wait a minute, I thought garlic was good for me. Well, it is, unless it's not. And so what has to happen is that we take dietary sulfur down, very low sulfur initially. And so Maria is one who wrote up this program, the diet program. And she, has created recipes and the whole thing to make it as easy as possible. We do that for about 2 weeks. And then we have people do a systematic reintroduction. So you bring in garlic first, and then you bring in onion, and then you bring kale and eggs, and each of these sulfur foods to see how somebody does with that particular form of sulfur. Because there are people who do fine with garlic, but they can't have a bit of onion. Or they do fine with broccoli, but they can't have any cauliflower. You introduce broccoli, no problem. Cauliflower, headache, or gut, or whatever. Because the sulfur compounds are very diverse in the world, in the foods that we're eating, and our body handles each of them in a different way, so we have to figure out which foods are okay for which people. And so that's a kind of a slow process, but once we get that figured out, then we have to get the diet expanded out as quickly as we can, because of course we need the sulfur compounds. We have to have them, but we got to have the ones that our body can deal with. Then also, so there's the issue of persistent toxicants. Another issue that I think is major with cancer is that microbes, in order to attach to cell surfaces, they have to break down the easy water layer. So they rely on that breakdown as a way of attaching, and specifically, I think mycobacteria are probably the most relevant in this regard. So that chronic infections become part of the drivers of that malignant process. They can be part of that, the driver in that process. So it isn't just that you can get somebody's sulfur working and suddenly the cancer is going to go away, because there are too many things that are happening here, and especially once cancer has been established for a long time, then the mutations do set in at a higher and higher rate, and as those cells become more mutated, then it becomes harder to reverse, what has happened genetically at that point. And at some point, you just got to kill the cell. You got to kill it. You got to just scoop it out with surgery or whatever. So I think that much of the importance here is with how we make sure that our bodies are processing sulfur adequately so that we don't end up needing to grow those factories into stage 3 and certainly not stage 4. So there's a quick summary, and I'll just put in a plug. I have a book. It's going to be coming out in about a month, I think. The Devil in the Garlic, and it doesn't just talk about cancer. There is a section on cancer, but also just about the role of sulfur dysregulation with all kinds of problems, and how does it happen that we get dysregulated with our sulfur metabolism, and then what do we do to fix it? So that's it. Thank you.
So, thank you very much for your excellent presentation, very interesting. Now, yeah, okay.
Thank you very much for your very interesting presentation. Is it possible that you put on the table all the references that were in your presentation, but they don't apply? You say there's many references, 1, 2, 4, 6. Do you have a picture of all of them?
I couldn't understand. Oh, the references, yeah. The next,
Thank you.
yeah, I've got 2 pages of them.
Thank you. One after each other so I can take a picture.
So yeah, so I'll put that up and then I'll...
Thank you, Greg, for this wonderful overview. But shouldn't we focus on shifting the battlefield from inside the body back outside the body? I mean, what you described is, okay, the last case scenario when you go look at it from a timeline. But if you bring it back outside the body, I mean into the microbiome, in our guts, in our intestinal microbiome, then by increasing our, let's say, microbiomal diversity, we probably alleviate all the symptoms of, let's say, sulfur incompatibility, or not possible to digest sulfur. Because what we are doing right now with our diet is we are clear-cutting our ecosystem diversity in our intestines, which is probably the major reason why we're coming into this scenario.
Yeah, so, and there are, you know, with the microbiome, and there's just crazy amounts of research coming out, and now there's like a gut-everything link, right? There's the gut-brain axis, and the gut-joint axis, and the gut-heart axis, and everything's got an axis to the gut now. And it's a catch-22 because you can't reestablish the gut, you can't establish health in the gut as long as there is a sulfur deficiency, because you have to have the bacteria. The bacteria of dysbiosis are what are helping to reestablish normal sulfur levels because they generate the compounds needed. Now, in an ideal world, we would never get into that situation. You know, we would prevent that from happening. But if we don't prevent it from happening, then those bacteria that we hate because they make us feel miserable are doing something really important, and we have to allow them to do that while we fix the problem so that they can go away. And in fact, just as a note in point, we have had a number of people with really miserable guts who have dramatic improvement just by doing Epsom salt baths. 4 cups of Epsom salt in a bath, Epsom salt bath. 4 cups of Epsom salt, hot bath, soak 20 minutes, 7 nights in a row. I mean, really crazy how people doing nothing but that will report dramatic benefit in their digestion. Why would that happen? It's not because it's not laxative effect. It's because Epsom salt is magnesium sulfate. Sulfate levels in the blood increase by doing Epsom salt baths as long as you have the concentration high enough. And that is sulfate getting in that doesn't have to come down the tube and meet up with those bacteria. So we're supplying sulfate with Epsom salt baths that then the body can use as a way of replenishing sulfate supplies, and you don't need the bugs anymore. So the bugs resolve and people feel better. So yes, if you increase the diversity of the bugs through fecal transplant, you can take care of the problem. Somewhat. What I would say is, if you haven't fixed, if there is a problem of sulfur metabolism that is more systemic, that even if you fix the bugs, they will undo themselves and get back to their dysbiosis, which we see all the time with patients who go through the protocols, they feel good, and there is a very common relapse after 2 or 3 months of reestablishing, of getting rid of symptoms because the more systemic problem hasn't been addressed. Yeah.
I have a practical question. if you want to buy food for your dinner, how you know which vegetable you select which is best suits for you?
Say it again.
So,
For the dinner, how do you select what is good for you?
How do you know what the best way to eat is?
yes.
Yeah. I'll give you my card. No, the way we do it, and this is, working in collaboration with Maria, I wouldn't be able to do it on my own. It's really, not everybody has a sulfur problem, but the people who look like they got a sulfur issue, and you can spot them, we just put them through the protocol. There's not a blood test we can do. We just put them through the protocol. We get their dietary sulfur down. We give them the nutrients that they need to process sulfur. We find out if they feel better by doing that. And if they do, we introduce foods and we find out what the sulfur foods they can eat. And then, once we figured out the sulfur piece of their diet, then it's just, their principles of healthy eating. We don't have like a particular diet that we do with everybody. Everybody needs to eat a little differently. And so Maria is working with people to figure out what their diet really needs to be. But we have to get the sulfur piece out of the way so that we know which of those sulfur foods can be part of their healthy diet.
Oh, sorry.
Oh. Yeah. Yeah. Oh, yeah. Oh.
No.
So quickly.
Yeah, no.
Take a picture quickly, and
We must
we have
prepare
to move on.
That's okay.
It's not working. We broke the thing.
We are running out of time.
Yeah.
Okay, so
There.
Oh. There's this thing.
Okay.
Sorry. Okay. I'm sorry.
Sorry.
yeah, that's enough.
Okay.
Pull the plug. I don't know if we're allowed to do this, but
our next presenter is Antonella De Nino, and she will speak about two fluids in one liquid water. Okay.
Good morning to everybody. And now we completely changed the vision of the problem because I'm a physicist, and this will be a presentation about the physics of water. I hope to be not too much boring for you. I'll try to do my best to explain my vision of water. Now, I want to talk to you about the two fluids in liquid water, and I want to talk to you about a hidden agenda. I think that everyone among you has his own hidden agenda. That is the fil rouge that connected all the action we usually do in our private life, in our professional life. And this is my personal agenda. This is what I'm trying to do in my professional life. And is to understand a two fluids model that has been foreseen in the past year by two Italian theoreticians, Giuliano Preparata and Emilio Del Giudice, more than 30 years ago. Many among you probably remember Emilio Del Giudice, who attended to the first water conferences since the Vermont time. And their theoretical proposal was very, very interesting, but I am an experimentalist, so my aim is to find experiments able to prove that that theory is actually true. So I'm looking for reliable and clear experiments because I wanted to I want to see if they are right. I believe that they are right, but my job is to prove it. And so I usually do experiments. And the experiments are aimed to validate their theory, even using different experimental approach. because the result should be to build a qualitative and quantitative validation of the theory. This is what physicists usually do. So, which is the basic idea of the Giuliano Preparata and Emilio Del Giudice? The basic idea, which applies to all the condensed matter, not only to water, is that the ground state of an ensemble of molecules condensed and in a coherent state differs from the ground state of an isolated molecule. One of the bases of the classical physics is that the nature is made up of separable particles. Each corp is separable by the other. This is not actually true in quantum physics. In quantum physics, it's possible to show that an aggregation of particles is submitted to different laws with respect to isolated particles. This is the starting point of the theory. So the basic idea is that the ground state, when I talk about the ground state, I mean the energetic state, that is the most stable level of the matter. The most stable level of the matter of an ensemble of molecules is completely different from the most stable level of an isolated particle. And why? Because if we want to describe in a complete way the matter, we have to consider interaction between particles and electromagnetic fields. Only in this case, we can attain to this new ground state, which is the real physical ground state. At this point, the dynamical evolution of the system is obtained in the framework of quantum mechanics, just coupling the Schrödinger equation to the Maxwell equation. This applies to water. What happened in water? If we apply these ideas in water, we found that under a certain temperature, that is 100 degrees, of course, and over a certain threshold of density, water molecules collapse in a coherent state. That is, they attain to a low level of energy, in which they acquire a great stability. These parts of the space in which molecules are condensed has been called coherent domains. Coherence domain, this is a pictorial representation just to fix the idea, but you don't have to imagine that in water there are such a cluster of condensed molecules like iceberg in the sea. This is an old-fashioned view of the system, because we are talking about a system in which the temperature and the pressure are the same. So from a thermodynamical point of view, it's a nonsense to imagine that in water there are clusters of different molecules. Thermodynamics teach us that whenever you have the same thermodynamical parameters, pressure and temperature, all the system is in the same thermodynamical state. So the coherent domains are not a thermodynamical state. They are an electrodynamical state. That means that there is a condensation, but not in the physical space, but in the space of the parameters, of the energy. So you have, it's not quite easy to see, but in water there are molecules which behave like a condensate and molecules which are free. Let's imagine a dense gas of free molecules in water coexisting with molecules which behave in a collective way. They oscillate all together with the same frequency and phase, and they have the same low level of energy, which give them stability. The energy gap which protects this particular arrangement of energy is quite high, and it's high enough to guarantee the protection against the thermal motion. So even at room temperature, it's possible to have this particular state of matter. There are other examples in the nature of this kind of states, or it's only a nightmare of theoreticians. No, we know that in nature we have the superconductivity and superfluidity that are well-known phenomena that are perfectly described by this physics. The only point is that usually it is said that, okay, it works, but it works only at low temperature. This is true, but it's true for superconductivity and superfluidity. If you do the calculation in case of water, you will discover that water is stable in a condensed state even at high temperature, at the room temperature. And so, in liquid water, we should expect the coexistence of two different kinds of molecules. Is it possible to see by eye in a microscope? Of course not, because it's not a state, a thermodynamic state. I can distinguish bubbles of gas in the liquid because they are two different thermodynamic states. This is not the case. So we have to invent some other methods to detect these two states if they are really. Now, and one thing to keep in mind is that these two states are, of course, related one to the other. They are related by a rule of sum. Their sum is equal to 1. So if one of the two increases, the other one decreases of the same amount. But they are related. This is the first formula that has been calculated shown by Preparata. And of course, the main dependence is on the temperature, but not only. Not only temperature changes the proportion between these two states. What's happening? Okay. It depends by temperature, but it also depends by the contact on the surface and on the dilution of solutes. So it's a very complicated mixing of different parameters which determines the amount of the condensed phase into the non-condensed phase or vice versa. Broadly speaking, just to not complicate it too much, the situation, we can say that gases are fully non-coherent systems. In a gas, all molecules can be described as independent. In liquids, there is a particular kind of coherence that involves only the electrons belonging to molecules. In case of solids, the coherence is extended to the nuclei too. So in this way, we can roughly have an idea of how the condensed matter can be described in terms of quantum electrodynamics. But which is the description of the physics, of the classic physics about water? Okay, we were told that in water there are hydrogen bonds. Hydrogen bonds connect one water molecule to another molecule. In liquid, hydrogen bonds, we were told, they constantly break and reform. And this gives the long-range structure to the water. The hydrogen bonds is a phenomenological concept that has been introduced by Linus Pauling in the 50s, and it's a very powerful concept. It was useful just to explain why the boiling point of water is so high, 100 degrees. It's very high temperature. It was impossible to understand why water boils at 100 degrees if you don't introduce another kind of bond, adding to the covalent bond of oxygen and hydrogen. But hydrogen bond, it's a very weak bond. The energy is very, very tiny. In fact, it is assumed that it forms and destroys every about picosecond, half a picosecond. And this is a very crucial point that we see later. What is an hydrogen bond? There are two criteria that I teach to the student at school. You need the first molecules as an hydrogen attached to a highly electronegativity atoms, and the second molecule has a lone pair of electrons on a small, highly electronegative atoms. So you need a hydrogen bond donor and a hydrogen bond acceptor. This is a definition. But what does it mean? Which is the lifetime of an hydrogen bond, which is the event which characterized the lifetime of a hydrogen bond. If we assume that, if we consider that the first violation of the hydrogen bond is the removal of the hydrogen, in this case, we will have a continuous lifetime. But if we assume that the hydrogen bond is violated only when one hydrogen is removed from one molecule and is attached to the other, this changes completely the situation, because in that case, you will have an intermittent lifetime. This is a big problem for my colleagues who are used to do a computer simulation of water, because depending on which is your model, which is your definition of hydrogen bond, you will obtain lifetime that spans from 0.1 to 10 or 12 picoseconds. This is a huge range, and this conditions very strongly the results of the computer simulation on water. In this way, you can obtain many different results. This is not good, as you can imagine, for physicists. But which is the physical background of the hydrogen bond? So let's imagine the electron cloud of an isolated molecule, as we can obtain by both simulation and measurement, is the one described on the left side. It's very smooth. It's a very smooth distribution. It's like a potato. There are no preferential directions in that distribution. So in that case, it's very hard to understand how a hydrogen bond can be obtained, because there are no preferential distribution. This is a problem. In the other, and on the left side, you can see the electron cloud of molecules which are collapsed in the ground state, which are in a collective motion. In that case, you will see that we have two directional preferential directions angled at 109 degrees. And those are the base, the physical base of the hydrogen bond. So we can imagine the hydrogen bond, but not for isolated molecules, only in case molecules have already attained to the ground state of energy. This is very important to understand what is water. So in the first case, no highly directional charge density required for H bond. So it's hard to justify the existence of water molecules arranged in long-lived hexagonal configuration. If the lifetime is 10 to minus 12 seconds, the probability to have an hydrogen bond is 10 to minus 12, which is the probability to have 6 hydrogen bonds living all together is the product of probability. So it's very hard to justify only with the concept of a hydrogen bond, the structure of water, the long-range structure of water. It's quite easy in the other case. If we assume that there is a transition of a part of molecules in a collective states, we can understand better what the hydrogen bond is. How can we prove this experimentally? By spectroscopy, mainly. Spectroscopy can be Fourier transform infrared spectroscopy, near infrared, or time domain terahertz spectroscopy, many kinds of spectroscopies. We have no time here to expose all the different spectroscopy that are possible to use, but just give you some idea about what happens in infrared. Infrared spectroscopy teach us what's happening with the OH stretching vibration. So it's very useful to study water. If I take the spectrum of a drop of water, I obtain a very broad peak, as you can see on the top. That is the spectrum derived by the vibration of the OH bond. It's a very broad spectrum, as you can see. So this means that there is not only one kind of molecules of H bond which absorb in that range, but they are very, very spread. And the spread, in this case, it cannot be justified only with the thermal motion and the statistical broadening of the line, but it's much more. So there must be something under that spectrum. How can you see what it is under? A trivial technique that is used by spectroscopists is to take the second derivative. If you take the second derivative of a line, of a curve, you obtain the minima corresponding to the maxima of the curve. If you do this for water, you can see that you have 3 minima. This tells you that there are at least and maximum 3 components under the broadband absorption of the OH stretching. And see what happened now if I had solutes to water. If I had solute, in this case, you can see these are chlorides, lithium chloride, sodium, potassium, and cesium chlorides. You can see that there is a deformation of the spectrum. There is a deformation that's very peculiar. On the right, the spectrum decreases when you increase the concentration. On the left, the spectrum increases when you increase the concentration. Of course, the total area remains the same. There is a peculiar point that is the one crossed in red, that is known as Isosbestic point, that is a derivation from a Greek word that means equal absorption, isosbestos, equal absorption point, that tells you that there is a point of equilibrium. Whatever it happens in the solution, there is a point which absorbs at the same way, independent on the concentration. And this is very interesting to study. Let's see what's happened, what we can learn from spectroscopy. If I do the spectroscopy, the infrared spectroscopy of solution of water, I can observe a deformation of the spectrum that is very intriguing, because the right part of the spectrum, which corresponds to low energy vibrations, moves towards the high energy part, and vice versa. The high energy part moves towards the low energy part. That is, that there is a change in the energy, but what is more important is that the change is in the OH bond length, because the maximum of the peak corresponds to the length of the OH bond. So there are molecules which are, when I had the salt in water, there are molecules which stretches and other molecules which shrinks. So there are two different classes of molecules which react differently to a stimulus, an external stimulus. The same can be observed even if you change the temperature. And if you want to have an example of this, you can look at a poster presented by Paolo Renati. In that case, it's not infrared, it's near infrared, but near infrared is just the first overtone of infrared, so it's the same. In that case, he changed the temperature, and the result is the same. So if I perturb water in somehow, I can clearly see that there are two classes of molecules belonging in a different way. So if I want to quantify this change, I can see that the area of the peaks, the area of the first peak on the right, always decreases, which whenever, whatever is the salts that I add to the solution. It's the same for lithium, sodium, potassium, cesium, and even other salts. The area of the peak in between stays almost constant. The area of the peak on the left, that is the highest energy peak, increases. This is what happened to the bond length. The bond length of the peak on the right decreases, and the other has an opposite behavior. So, since our system is the superposition of two components, where the sum of the density is conserved, we can understand the real meaning of the isosbestic point in water spectroscopy. The molecules belonging to different populations react differently to the solution of ions. So this is a case in which we can observe the presence of two separate classes of molecules in water. And this isosbestic point is the equilibrium point between the two fractions of water. So, this implies that one population is depleted whilst the other is increased, and the crossing point between the two fractions divided the region where water-water or ion-water interaction prevails. I show you that there is a class of molecules which shrinks. That means that in presence of a perturbation, the interaction between molecules in the first population, let me call first population, is higher than the interaction between the molecules and the external ion. So the molecule prefer to stay closer together. In the other case, the interaction with ions prevails on the interaction with other water molecules. This completely different behavior. So we want to do an assignment of this population. Let's observe, the red curve is the spectrum of water. The black one is the spectrum of 3 molar sodium chloride. And under the spectrum, you can see the convolution in 3. Now, if I observe what happened on the right of the isosbestic point, you can see that the right side is almost coincident with the lowest energy band, sub-band, under the spectrum. And the other part, the part that is at higher energy with respect to the isosbestic point, coincide with the high part of the spectrum of the other two components. So it's possible to identify molecules below the isosbestic point with a low energy fraction and molecules above the isosbestic point with a high energy fraction. What we can expect from coherent domain theory? So, calculation tells us that coherent fraction has a lower density, and in fact we observe a higher molecular dimension in the lower energy molecules. So it's okay. The calculated energy gap between coherent and non-coherent fraction matches the value of energy difference between low energy and high energy fraction measured with vibrational spectroscopy. I have no time to show you the details, but again, in Paolo Renati's poster, you can see how it's possible to calculate the energy difference between these two components. And then the low energy coherent fraction is depleted by the temperature and by the addition of solutes, which have a Cow-trope action. And this is exactly what is predicted by the theory. Interaction among molecules in coherent fraction is much higher than interaction with the ion in solution. So again, molecules in the coherent fraction actually have a very strong interaction among themselves. So this is confirmed by the experimental results. Now, let's come back again to the isosbestic point. The isosbestic point can be seen as an equilibrium constant. In this case, I can take the area of the red part over the area of the blue one, or vice versa, it doesn't matter, and say that this is the equilibrium constant of my systems. What happens if I want to study the equilibrium constant of my system during a perturbation? As example, when I had solutes in my water. Okay, this is what's happened. I defined, it's arbitrary, the same is the opposite, of course. I defined the ratio of the blue on red area, and I plotted with respect to the concentration. And this is what I got. These are sigmoid. That means that the system is structured in two levels. Below a certain concentration, nothing happened. Above a certain concentration, there is a restructuring of the system. Until the second plateau that is not technically reachable because there is a precipitation of the salt. There are some experimental problems, but there is a second plateau on the right. The second plateau means that nothing more happened to the system because everything has already changed. Change how? It's changing this way. If we look at just the linear part that connect the two plateaux, we can see that in a log-log scale, it's a straight line. Those straight lines, all the straight lines, have the same slope. The slope is the same for all the ions. That means that there is a property of water and not of the ion. And what does it mean? If I have a straight line in a log-log plot, this means that there is a scale-free law that connected the two y and x axes. Again, what does it mean? This shows that our system has a self-similarity property. That is, that water rearranges in a fractal way when it's under a perturbation. And this is very important. You can see the same in infrared, in near infrared, it's okay. What is very important is that the power law and the fractal behavior is not a simple way to define the organization of a system. It's a trademark of an auto-organization in a complex system. It's completely different. It's not possible to pass from a linear organization to a fractal organization. They are completely different. And this is what we have learned in mathematics, the difference between exponential and scale-free network. In a scale-free network, is it possible to have some hub? That means that one node of the net is connected with many. And this is exactly what happened to water, especially in the biological realm, in which water and a few other molecules behaves like a hub, can develop many relations with other participants, while the most of molecules in our metabolism just do the reaction with only other one. Water can make reaction with many other molecules in the biochemical path. So the auto-organization is the result of a dynamical process that is based on the interaction among components, carrying out a configuration having properties different from the original one. It's the same difference which is between one bird and a flock of birds. The behavior is completely different. So if we add to this that Giuseppe Vitiello, another Italian theoretician, has shown that the auto-organized systems are described by the same mathematics which describe the quantum coherent states, we have a clear picture of what are these two fluids, two components in liquid water. I finished my time, so I thank you.
Thank you very much, Antonella. Sorry, we had to rush you a bit, but it was a very, very interesting talk. We have time for just one or two questions, maybe?
Yes, and then an announcement after questions.
And then there'll be an announcement. So.
Thank you very much, Antonella, for just a very, very interesting and beautiful talk. What do you think, is it possible to include in your model ortho and para waters? That water, ortho and para water. It's now very well known that...
No, I've never thought about it. I'm sorry.
Yeah,
Not yet.
Yeah. Okay, thank you.
Antonella, thanks for a beautiful presentation. As you know, water structure tends to build next to certain, particularly hydrophilic surfaces. You didn't mention that. I'm just wondering, is there an easy way to explain how that would happen according to your theoretical presentation?
An easy way to see what? Sorry. How?
How it is. If you can somehow explain how easy water can be formed
yes.
according to
Okay.
this.
I'm very sorry because I prepared a few slides to show how easy water can be included into the picture, but I was too long. I'm very sorry. I had no time to show you, but if you are interested, of course I can.
Thank you. That was brilliant. Roentgen proposed two types of water molecules, what he called like ice molecules, which are less dense than water molecules, and that they somehow changed as temperature changed. And he used that to explain like the 4-degree maximum density. Do you see anything in that? Is this model that you're doing, will that help explain perhaps some of the anomalous properties of water at different temperatures?
The nanostructuring.
Pardon?
Sorry? Can you? Anomalous structure.
Anomalous. Yeah. Are you familiar with Roentgen's proposal of the two ice molecules and water molecules?
I'm sorry. I can't.
Oh, sorry, the language barrier.
Can you speak slowly, please?
Yes, Roentgen, the old scientist, I can't remember what year, he proposed two types of water molecules, ice molecules and water molecules, both contained in the fluid at the same time, but that the relationship would change with temperature.
Yes, but the problem, the main problem is that you have to decide if you are talking about thermodynamical phases or not. Because in case they are thermodynamical phases, the two kinds of molecules are described by what? by pressure and temperature, because the first model of two fluids in water has been proposed by Röntgen at the end of,
Yeah, that's what I was talking about.
Yes, okay, but the problem of Röntgen model is that he imagined something like iceberg in a liquid. But iceberg in a liquid are not possible, because if the liquid has the same temperature and the same pressure, you cannot have two different thermodynamical phases. And this was the reason why the first proposal, the Röntgen proposal, was dismissed by the scientific community, because the objection was had a thermodynamical base.
Thank you. Okay. Thank you very much, Antonella. And then I think we have an announcement.
Well, is this announcement? Yeah.
So I have a few announcements about the further program. So first of all, after lunch, you can see in your program Bad Southern City Tour. I would just like to let you know that there is no organized tour, so it's like self-guided tour. You can just take a walk. We have a few maps at the table of registration, so you can take a map and see where you... Okay, so next announcement is about time change. So German time would switch this night 1 hour back, so please be sure to adjust your clock or your phone so you have an extra hour to sleep tomorrow morning. That's the good news. And then also after lunch there's going to be a workshop, and I think Hans is going to tell you about that.
Thank you very much. A little bit description, more information about the workshop, which means we have the supporters outside with a window of 15 minutes. Every supporting company outside will present their product, which is very interesting because how is being science converted into products that are being used is a very interesting thing. Yeah? So how is all this, what we are doing since years here, being realized in the real world out there? Yeah? That will be an interesting thing, and everybody will have 15 minutes to describe. We hope that a lot of questions are coming out, and parallel to it is the little exhibition out there where you are welcome to go and visit and ask questions. Yeah? Thank you.
Okay. Oh, and one announcement. The dinner tonight, the banquet, would take place here in the same room of the hall, not in the dining room. They will change the table, so yeah, please be sure to come here. And now enjoy your coffee break. See you later.
Let's try and come back for 11:20. That's possible. 11. you got? Okay, everybody. Okay, shall we start? Or try to start?
Germano.
Okay, can I invite everybody to sit down, please?
Okay, so you see. Okay.
It's still out there.
Yeah. We are starting.
Exactly. Please sit down so we can start.
Will you introduce him or? Okay, okay. So please be seated. And our next presenter is Roberto Germano from Italy. And he will speak about homochirality of, this is,
oh, sorry.
yeah. Interaction among water and magnetic fields. Okay.
Thank you. Thank you very much to the organization, to Jerry Pollack, to have invited me to share with you this question mark. This is a question mark, okay? So, I want to briefly present myself. I founded in 1997 this spin-off company of the National Research Center in Italy. And we are performing technology transfer, connected in particular to physics, applied physics, especially in these three areas. The idea of technology transfer can be this one. You know this person very well, and this year, it's since 500 years ago, he was dying. He was doing this drawing. This drawing is something that was disappearing for 400 years and then invented again. The ball bearing. This is an example of missing technology transfer. So we try to avoid that in the future something like this again is happening. Okay, we are staying in Napoli, you know, South Italy part. And also we have a little office in Berlin. This is our team, physicists, engineers, and so on. We are a micro company, and we are facing with something that you know very well. Every day, no? This is our common problem, and we know very well this problem. In effect, this is another example, no? Normally we say, think outside the box. In effect, we have to try to think like there is no box. Okay. So, the question mark is this. The problem is that the origin of so-called bio-homochirality is still inexplicable. It is the homochirality in the bioorganic world. I am a physicist. Many of you are biologists, so I understand much more than me this topic. So, exclusively left enantiomers of natural amino acids are found in nature. Exclusively right enantiomers of natural sugars. And we don't know why. In general, many molecules in natural biosystems have been found to be chiral and to have all the same chirality. So, we don't know why. You know, everybody knows the origin of the word, but I like to speak about the origin of the word in general. Remember that chiral is coming from hand, because in ancient Greek, this is the name of the hand, because the hand our ends has this characteristic. The mirror image cannot be superimposed. In nature, we have many examples of chirality, like this kind. So there are kind of preferred handedness as a common trait of living things. For example, this kind of plants have these characteristics. Convolvulus arvensis winds to the right, for example. Lonicera semprevirens winds to the left. Like snails, such Ligus virgineus, are generally right-handed, but within a species, left-handed versions appear owing to mutations, for example. Or in bacterium, Bacillus subtilis normally forms right-handed spiral colonies. When heated, this change to left-handed. Obviously, also atoms and molecules can have these characteristics. So be asymmetric with respect to left and right. But normally, we don't know any connection with living things. Let's consider water molecule. Water molecule is not chiral at all. It's symmetric. So, this is the end of my talk. or isn't it so simple? This is the question. Okay. So this is a table that published in which we can understand what is the left and right chiralities. The gray areas are most common. The white areas are less common in nature. So there is this kind of characteristics. Okay. Another topic, very interesting, is connected to magnetism, the origin of the puzzling origin of the magnetic field sensitivity. Almost ubiquitous in living systems, animal and plants, from unicellular to mammals, deserves to be phased up, thanks to new ideas. This is another problem, okay? So, this is a very recent paper that I found, in which they are showing that geomagnetic fields are sensed by human brain. Okay, normally one thinks that the mechanism is connected to little magnetic particles inside the cells. This is typical, in which everybody try to explain this. Okay, so this is very interesting. Also humans have this characteristic. This is a very recent paper. So, despite being aware that our insights are at the preliminary stage, I'm presenting a short talk about how mirror symmetry breaking in liquid water may contribute to the origin as well as pervasiveness of bio-homochirality, maybe via the Earth magnetic field influence. So, it is well known that water molecules constitute more than 90% of the whole number of molecules entering living organisms. I'm speaking about number. The representation of liquid in quantum field theory shows, as Antonella De Nino was explaining very well and very clearly before, shows that in the ensemble of molecules show a new minimum energy, where a fraction of molecules is forming so-called coherence domain, surrounded by an uncorrelated ensemble of molecules. So, as Antonella De Nino was showing to you, this is a kind of picture of the liquid, in particular of water. By water is particular in this respect, because from QED calculation, the result that the energy of the excited states is 12.06 electron volt, which means that only a small amount of energy, 0.5 about electron volt, is sufficient to release an electron. This is very important. So this means that, Okay, this is the situation, the class. A normal isolated water molecule cannot do this at all, because the energy is a lot. 12 electron volt doesn't mean X-ray, doesn't mean it have very many thousands of degrees of temperature, so it is not possible. But we know, and biologists were the first to understand, that liquid water has a lot of electrons inside. It is not insulator. So, considering that the coherent state is the superposition of these two molecular states with this different statistical weight, this means that at each instant, 0.13 quasi-free electrons per molecule are present. That is 700,000 quasi-free electrons for each coherence domain. There are a lot of free electrons. This implies that coherent water is a reservoir of quasi-free electrons. Moreover, water CDs have a non-trivial internal energy spectrum, so that there is the physical possibility, due to the natural energy minimization processes, of the onset of coherence among spatially separated CDs, giving birth to a phase matching in extended region of liquid water among the electromagnetic modes and the matter component of an extended space-time region, succeeding in this way to connect microscopic phenomenon to mesoscopic ones. The picture is something like this. You have a zoom, no? A coherent domain, many coherent domains in phase, and so you have extended region of liquid water that are, in a certain sense, organized, as Antonella was showing before. So, in this, and I was speaking about this in this paper, if somebody is interested more deeply in this topic. So, now let's go to many experimental facts. As Vittorio Elia was showing to you 2 days ago, he was evidencing, his group was evidencing for the first time micron-sized chiral supramolecular H2O aggregates in water at ambient conditions. We were working together in these years. I'm so happy that I can work together him and together Elena on these topics, because when I was a young student, I was following his first talk at physics department, and I became crazy about these new ideas and new experiments. So what is the meaning of this? That the chiral configuration of this water-organized structure are experimentally evidenced by spectropolarimetry techniques, by their effect on light polarization chains, the so-called circular dichroism. This technique is evidencing this phenomenon. So it was published on Water Journal, and these are some images of these larger aggregates that Vittorio Elia was explaining to you 2 days ago. Okay, so we see this is the CD spectra, showing in particular a beta sheet behavior of this kind of aggregates. How these aggregates were generated? They were generated by physically perturbing pure water. For example, iteratively immersing a hydrophilic membrane, like Neptune, in milky water, stirring the liquid, removing and drying the membrane. Like this. This is the action, the method. But it is working also with many other hydrophilic polymers and also other methods, not only this method. So it is a very common behavior. So what is happening? You are starting with pure water, milky water. You are doing physical perturbation. You have large changes in physicochemical parameters, pH, atom mixing, electrical conductivity, viscosity, and so on. You are performing all the analysis, but it is still pure water. But it is showing circular dichroism, typical of macromolecules, beta sheet, and also UV fluorescence in the same range that Jerry Pollack is seeing in the exclusion zone. Something new, it is not typical of H2 molecules. And this is the beta sheet resembling, okay. This is the typical spectra. Moreover, it is happening something crazy. If you are lyophilizing, as Vittorio Elia was showing to you 2 days ago, there is a ponderal solid residue, a lot of solid residue, that you analyze and you don't find almost nothing. Sometimes you find some carbon. It depends on the origin, on the method, but I cannot now. This is interesting. We were calling this Zedrill from ancient Greek xeros, dry, hydro, water, ile, matter. Because the important to understand that it is a class of material. It is not one molecule, not one organ, not one material. It is an entire class of new materials that are forming from this kind of methods. And this is the fractal appearance of this matter. You can see the different length, 1 centimeter, 100 micron, 1 micron. In particular, this is obtained by AFM, and you can see that the spheres, the little particles are nanometric, like nanometer, 1, 2 nanometer in size. It is interesting that a Spanish group was theoretically calculating that if you take a piece of exclusion zone, it is energetically favorable that this piece is organized in a spherical structure that is showing exactly that fluorescence that we see. We, Vittorio Elia group, and also Pollack and so on in exclusion zone, the same fluorescence. So probably this is what is happening. A piece of exclusion zone is detaching, it is organizing in nanostructure, and the nanostructure are also organizing in wires and so on. This is the structure. This is interesting to see because this is a similitude, no, among protein and this kind of structure. Only similitude, okay? So, we showed that the aggregates forming in water, adjacent to a hydrophilic compound, may reach and sustain a stable chiral molecular configuration. And this finding was confirmed after a few months by the first observation performed by another research group of a chiral water superstructure surrounding DNA at ambient condition, revealed by second-order nonlinear optical spectroscopy. This is an experimental work. It was published a few months after our findings. Exactly, very, very interesting. So we were writing a little communication to say, okay, let's try to put together this information, because in effect, the interesting thing is that we were noticing this without DNA, in absence. This is our publication in Journal of Molecular Liquids. Now, let's start with some theoretical clues. Together, a theorist now retired from CERN, Claudio Verzegnassi, from Trieste in Italy, we were trying to go deeply in the problem of the quantum field theory treatment of possible magnetic effects on a system of free electrons. This was published in Journal of Magnetism and Magnetic Materials 1 year ago. The main result of this paper is that if you have a system of free electrons, when the magnetic field is inverted, the change sign, change the energy of the free electrons. This is not classically Forresting at all. This is a new result, thanks to the theoretical capacity of Claudio Verzegnassi in particular. Okay. So, It is interesting, this clue, recent paper published in Journal of Physical Chemistry B. You will, the author will speak tomorrow, and it was publishing in Water Journal also, if I remember well, something. And it was publishing in Journal of Physical Chemistry B 1 year ago the effect of spin polarization on the exclusion zone of water. And it has been shown that a right-handed magnetic vector potential can enlarge the EZ zone, while a left-handed potential showed no potentiation, but sometimes a depression effect on the exclusion zone of water. So this is another experimental clue, very interesting. You can ask to him, he will speak tomorrow. So, a conclusion is that changing the sign of the magnetic field, the effect of the energy of the Z water change sign. But this is exactly what quantum field theory predicts for a set of free electrons, like I showed in the preceding paper journal. Another experimental clue, the role of the electron spin polarization in water splitting. This is also a very recent paper. It seems that there is a role in water splitting on the electron spin polarization. Okay. So what it is, let's go to the question mark. There are a lot of clues, no? So once is happenstance, twice is coincidence, 3 times is enemy action. Probably there is something like this. In effect, Jan Fleming was famous also for another, yes, you know, my name is Bond, hydrogen bond. So, how to convert the quantum field theory prediction for free electron into measurements? This is what we want to do. The idea we decided to follow and to check is the following. Together, the theorist Claudio Verzegnassi, is this. Given the fact that easy water has so many free electrons, its reactions to a magnetic field should just be the reactions of its free electron components. The latter one can be computed and would provide theoretical prediction for the Z water reaction to be experimentally measured. So, what is the choice? What are the energy and chirality? Which it is the observable that we want to choose. So, together Claudio Verzegnassi, we wrote a paper that was submitted to Water Journal, and it will be published, I understood, in a short time, after many months of discussion with the referee. And this is the paper. Magnetic effects on the chiral nature of free electrons in quantum field theory. What we see is this. A magnetic field is able to modify the polarization index of an ensemble of free electrons and their chirality features. So, now we want to do the following experiments. It is important, with all the experimental clue that I always showed to you, it will be fundamental to understand if the right or left-handed chirality of water, supramolecular aggregates, has or not a preferential side in ordinary conditions. This is not so easy to measure because, as I told to you, there are many kinds of aggregates together in liquid, and when you have not a pure molecule in liquid, it is not so easy to perform this measurement. I discovered, because I am a physicist, I was thinking, okay, it is very easy to do this. But I understood that it is very, very difficult when you don't know the exact molecule, one pure molecule in the liquid that you are measuring. So it will be a problem, but it is very, very interesting. And afterwards, to measure if their distribution, left and right, that can be also 0, is changing under an applied magnetic field. So, but what is the connection, repetita iuvant? This last physical possibility to connect microscopic phenomena to mesoscopic ones is opened via the non-trivial internal energy spectrum of water CDs. This is the connection. Otherwise, it should be impossible to think about this, no? This is the problem, a fantasy. Okay, so I dedicate this talk to the living memory of my friend and teacher, Emilio Del Giudice. Thank you for your attention.
So thank you very much for your excellent presentation. And now let's see some questions.
Thank you very much. An excellent talk. Chirality is so important for what we're looking at. At WETSUS, we are showing that bacteria are incredibly important for understanding how carbonates come out of solution in water. And of course, if bacteria are involved with the process, they will introduce chirality into the structures that are formed. So I would suggest that it's very important in these experiments that you ensure there is absolutely no living matter whatsoever in the water that is being used to then explore the chirality of what comes out of solution.
Thank you for the suggestion. We are collaborating now with a biology research group who is involved exactly in this topic, because it is, in effect, very important. At the beginning, we were not understanding, we are not thinking about this. So it is really, yes, something that we are facing just now. Thank you. Thank you very much.
Thank you. I am not physics, just therapist. And I was hear your presentation, and in the beginning I thought that it will be technical, too much for me. But some point it's very interesting from my point of view. You said that the distribution of chirality, right or left, is changing under applied electromagnetic field.
No, it is a question. It is a question.
It's a question?
Yeah. It is not sure. We have to understand. There are some clues. There are some clues and some theoretical predictions. But this is what we want to understand. Yeah.
Okay. So, if suppose is changing for field or for another forces, what do you think it will be influence on the water? Its properties or its state, or what kind of change you waiting for or you looking for? Yes,
something like to use a metaphor, like a matrix. If a magnetic field should be like a matrix for what is happening in the matter world, because waves matter, to do a joke, in the sense that there is this kind of correspondence. So something like this, via the dynamical richness of the coherent domains in water. So there is an internal structure of water. It is not a chaotic ensemble of H2 molecules, no? Because if you see liquid water as a chaotic ensemble of liquid molecules, it is impossible that a magnetic field can do something to this kind of... Also, it is really impossible. But it becomes probably possible via this connection that I showed in this talk. Also in part are only experimental, and there are some clues that are theoretical. So it is nothing so clear. We are trying to use also these clues to try to understand something that until now seems not understandable. Yes. Sorry.
Well, thank you, Roberto. I was wondering whether you have any indication or clues as to the higher order of the simulated MD structure that you presented, how that could assemble in a chiral manner. like in a three-dimensional structure, that would be chiral. And the second question I have is whether there's any indication that ice could form chiral substructures, because thinking of the analogy between H2O and SiO2, silicon oxide, it is known that quartz crystals form left-handed and right-handed macroscopic crystals. So could one of the structures of this XY2 molecular framework be superimposed onto ice? And is there any indication that ice or even a higher cluster in water could be chiral?
I don't know.
Oh.
really, I don't know. But we have to try to understand also this. Yes, we can speak because I think that we are beginning to collaborate. So it is interesting to try to understand also this. Really, I don't know.
Thank you, Roberto, for the presentation. Very interesting. Just a question, and half a question and half a comment. When you're thinking to see if whether the magnetic field is able to affect the chirality of water, of course, of this superstructure of water. I was wondering, maybe to establish whether it occurs or not, we have to create a sort of symmetry in the system, in the sense, an axis of symmetry with respect to something change, in the sense, aggregates in water that are forming are immersed, for example, in a static field that is oriented upwards. But of note, there are some aggregates that are rotating and whatever, so you should introduce also another elements that break the symmetry with respect to which you have the up and down.
Yes. No, in the theoretical paper that will be published on water in the next, I don't know, weeks, months, you will find the calculation performed by the theorist Claudio Verzignassi.
Yes, I know him. Yeah.
In which it is showed this breaking of symmetry.
Okay.
Yes,
and
this is very interesting.
In fact, were you thinking only to static magnetic field or also to alternative?
Sorry.
Were you thinking to only static magnetic fields or also to?
No, in the theoretical paper, we are only using a static magnetic field. But who knows? Probably.
Do you think to operate in a metal chamber?
Probably it is possible that not static magnetic field can do something. This is all just the beginning of this.
Yes. But are you thinking to operate in a metal chamber or exposed to also the natural fields?
I don't know. We will organize. We will organize the experimental setup.
Okay, it's just a hint.
We will speak.
Yes, it's just hints. Thank you so much.
Thank you. Yes, very interesting idea, of course, and it seems to me that we should be reminded that most interesting things happen at the surfaces. And water air surface is just a kind of easy water.
Yeah.
So their water should be already somehow organized from the electronic point of view as well. So that is one condition. And there is another condition. There is a geomagnetic field, which is a constant and also variable. So we have tremendous surfaces, if you just to look at Earth, and the tremendous effect of geomagnetic field on these surfaces. And in fact, I found probably many years ago, but I didn't realize that water is chiral. And everybody can get this, come to this conclusion. If to look at the solubility of L and D amino acids, depending on temperature, it turns out that they are different. Solubility of L and D amino acids in water, depending on temperature, is different. If water was achiral completely, it should be the same.
Very interesting. This is another very, very interesting clue. Really. Thank you.
One last. Could it be information that makes a difference here?
Next. Sorry. I was not.
Could it be information that makes the difference?
No, I was not understanding the question. Sorry.
If it could be information that makes the difference. I mean,
Information.
In chirality, information. So it was.
I don't understand very much the question, but
Yesterday we learned that information can make a difference of matter. We saw a presentation yesterday.
Yes. Okay. Yes, it depends on what is the definition, how we define information. In effect, information, the origin of this word is in, to put in form. So we are speaking about shapes, to put in shape, no? Information from Latin, to put in shape. So we are speaking about shapes, so in effect, probably there is a connection. The etymology is all the time very interesting. Okay.
Okay, so thank you again.
Thank you to you.
And represent. Thank you. I will do this. Okay. Thank you.
Yeah, so I was going to present our next speaker, who is going to tell us about large-scale structures in ionic solutions. So, oops. Yeah. Yeah. Okay, thank you.
Good morning, everybody. My name is Chud Yi. I'm going to talk about the large-scale structure formation in ionic solution and its roles in the electrolysis and conductivity. The starting point for this investigation is observation by Professor Pollack, reported in the 4th phase of water book, that when the water is being charged with 2 electrodes, the zone of alkaline zone and acidic zone will form. And also from Pollack Lab, a 2009 paper, that state that the zone are also charge bearing, and as much as 70% of the input charge can be obtained back by discharging. So this is this is a tube of sort of salt water here, and then in progressing through time, 45 minutes, you can see the acidic zone and the alkaline zone forming, and the meets in the middle. So what I'm trying to do is to find out what really happened inside inside the zone here. What is the picture inside the zone? So I constructed this setup here, which is just a long tube of water, 2.5 cm by 2.3 cm of water height, and 34.5 cm of length. The long tube's configuration is to constrain the charge zone formation so that they come from both ends and the meets in the middle. And why 2.5 cm? It's just a guesswork, and it just turned out that it works quite well for me. And I have 15 probes in the middle. These are just platinum wire shielded with the PVC cladding, with just the tip exposed. So they're dipping into the water, halfway through down the water depth. The electrode is platinum electrode, 5 mm at 26 gauge. The size and the shape of the electrode is secondary important here because they determine the rate of electron, the rate of electricity flow. So for our experiment, I limited down to below 10 milliampere because above that, there's a lot of turbulence and too much bubble forming and so on. So that disrupts the charge zone formation. The control circuit is given in this schematic diagram. It's just 2 electrodes connected to a power source. And there is a power toggle switch here, polarity toggle switch. So that means the charging can be flipped, to positive charging and negative charging. And there are, there is a switch here that actually connects the electrode to the power source, or can disconnect the 2 electrodes so they stay in the open circuit mode. And the switch can also switch to here for discharging. And there's a digital multimeter sitting here doing measurement. And there's a switch here that actually sort of can open the circuits and to route the current through the circuit. Otherwise it's just the multimeter is measuring the voltages. And then the 15 probes here are connected to a multiplexer. So there's a control circuit there that sort of goes through the measurement. The measurements are all done with reference to the electrode B. So that's how it looks. This just there's a computer control relay with a spaghetti mess of wires. And a program doing the controlling and logging of the data. Okay, now this is the profile, the voltage profile of charging 20 millimole Na2SO4 at 10 volts for 6 hours. You can see here that there is a drop of 2.5 volts at both ends, and then at time 0, and then the voltage are spread out through the electrode. But at the time scope 1, you can see that some of there is an upward climb in the voltage of some of the top 9 probes, and then the bottom 6 probes sort of drop down, and there's a big gap forming in the middle. So if we plot the time shot profile of that t equal to 0 and t equal to 3 hours, 180 minutes, this is what we get here. That is the probe position versus the voltage. So at time equal to 0, we see that there's an initial voltage drop, and then the straight line. But at time 180, we can see that there is, the 6 probe has sort of taken a negative bias, and the top 9 probe has taken a positive bias. So, but what is the voltage at the probe position? We don't really have a reference point. So what I did is that I cut out the power source temporarily, just for about 2.5 seconds, and then I did the measurement of all the probe position. So this is the profile of all the probes without the power source. The water is still charging. I mean, it is just a temporary 2.5 seconds sort of cutting out of the power source to do the measurement without the power source. You can see that that is probably where the 0 line is, but then the 0 line is also drifting. So in this configuration, there is no absolute sort of reference point for doing measurement. Because everything is all relative to electrode B. You can see, the nice picture here. The probe 14 sort of begin to take on the negative bias, and then followed by the probe 13 and so on. And at the bottom end, there is a P0 and the P1 and P2, P3, P4, and the P5. You can see, that the probes are actually sort of split into the positive sides and the negative side, with the 0 point somewhere in the middle, somewhere here. So we can see the drift here. You can see that there is a kink here. There's a point that the voltage started to drift outward. You can see the kink, the progression of the kink. And the time scale is really slow. That is 3 hours and the 6 hours. So it's a really slow process. So the building up of the zone, the charge zone is a really slow process. Okay, now after charging for 6 hours, the water is left to sit in open circuit mode. So there's no power source anymore. We can see that there is something happen, after about 3 hours. Now so when that graph is plotted with reference to, if I switch the plotting reference to problem P1, that's the picture I get at the bottom. What that is saying is that the P1 actually sort of hold a rather constant voltage. So they mean it's actually useful to be used as a reference point. But then you can see the VB here, the charge zone, and there is actually a charge at the electrode B, high negative charge electrode B, and then that one here collapsed suddenly. Now this is actually tracked over 10 hours. You can see that the voltage is actually holding really, really well through the 10 hours. This is really surprising. Now we have a situation where we have a zone with negative charge and a zone with positive charge, and they're sitting next to each other and they're not mixing. What I suspect is that the negative charge zone has a layer of EZ surrounding it, that's insulating it from the positive charge zone. And that is why it is the two zones are holding apart, for extended length of time. So we can see there is a sort of quantized nature or discrete nature of the charge zone. You can see the VB here holding constant, then a sudden collapse, down, then it's hold constant again. Whereas the positive charge zone are not so quantized in nature. You can see the VA sort of decreasing, it's gradual decreasing. But nevertheless, there are 9 probes here that are actually sitting at a rather constant voltage here. So that means that there is the negative charge zone has shown a very strong sort of a quantized discrete nature in terms of the charge density it can hold, but not so much for the positive charge zone. Okay, this is the charging of the 40 millimole HCl for 6 hours at 4 volts. So this one, after charging for 6 volts, I leave it in the open circuits and to track the voltages. We can see here there is now at the negative end, we can see the 3 quantized level here. There's a VB, there's electrode B sort of drop down to that level and stay constant. Then P1, P4, and P7 sort of stay at one level. And then P0, P2, P3, and P5 stay at another level. So there's 3 quantized levels on the negative charge zone. And all the charge zones, they hold the charge, more or less no dissipation over the 1 hour interval. Whereas for the positive charge zone, we can see that they're all drifting towards some sort of a common voltage up here. So there's a sort of incremental drift. But the negative charge zone, they hold the quantized sort of voltage level. That is the current for charging of that's the same experiment for NS2SO4, the 20 millimole NS2SO4. We can see that there is the voltage rise from 4.4 milliamp to 5.3 milliamp. That is a tremendous rise in the current. And that rise actually corresponds to the building of the charge zone. So that means all through this period that there's the currents are rising. But then you can see that at this point something happened here. All the voltage of the positive one started to drop down to a level. You can see there's a corresponding. That is that point here. Now, when I reverse the charging cycle, we can see that the current actually dropped down to where the original level, almost the original level, and then it starts rising again. So that means that the rise in the current is not due to any electrolysis byproduct, accumulation electrolysis byproducts. It is actually sort of due to the charge formation. Now, this is an interesting case. This is 1 millimole Na2SO4 charging at 20 volt. That is a really low ions concentration. And that is more or less the threshold sort of ion concentration where the charge will form. There is a minimum concentration in charge density for the charge formation. And when we can see the current from 1 to 2.5, that's 250% increase in current. That just by virtue of the charge formation. At that point, I reversed the cycle. We can see that the current has just dropped down and then so it dropped down to the initial level and never rise again. That is the charge run figure here. We can see that the forward cycle, there's the charge run formation, But then on the reverse cycle, we can see the dissipation of the charge run, and then it just dropped down to, there's no random noise level. And we can see that there's also correspond to the current dropping down here. So that means that the charge run formation has an enormous effect in the conductivity of the currents in the water. We are seeing a 250% increase here. That is the charging of the 40 millimole HCl. We can see the current actually sort of drop down. So that means that the effect of charge formation is not just increasing, it can also have the decreasing effect. But also take note of how jagged the current is. So what that is saying is that what that is showing is that there is actually some sort of insulation level, some sort of insulation happening somewhere, and I believe it's at the negative electrodes, and there is some sort of breakthrough event. So that means that there's a burst of breakthrough and there's a jump in current, and then another burst through, another sort of breakthrough, and there's another jump in current. So then sometimes the current can be smooth. When it's rising, when the current is rising, usually it's smooth. When the current is sort of decreasing, usually it's jagged. So that means that some insulation that need to be breached for the current flowing, for it to flow. So now the question, if the current can vary up to 250%, so what about the oxidation or reduction potential, and the electrolyte strength of the ion? So, because all the oxidation, the ORP and the electrolyte strength are all measured in water environment, aqueous environment. So if the current can vary by such an enormous amount, so shouldn't we sort of take the effect of water into the measurement somehow? So there's a question. Now, the deionized water is another interesting case. We can see that is actually showing that is the forward cycle, a reverse cycle, forward cycle, reverse cycle, and forward cycle. So there is 1, 2, 3, 4, 5, 6 cycles of alternating sort of charging, and the current sort of increase from 3 microampere to 6 microampere. So the deionized water can be considered as the water with some sort of trace amount of impurities, and also including the dissolved CO2 and so on. So there's some level of ions in the water. Now, the interesting picture is this one here. The voltage is jumping all over the place. It took me a while, you know. I was looking at this picture for one whole year before I realized what happened. What I think happened is that there are small charge zones forming at electrodes, and the charge zones are sort of forming at both electrodes, and the charge fragment are migrating across the whole length, landed on the other side. That's causing a burst, you know, a sort of burst in the voltage. So that means that the mode of transfer of conductivity is actually through the charge zone formation at the electrode, and then the charge fragment sort of travel to the other side. That is, you know, because the swapping of the charge fragment is causing the big jump on the probe voltages. So what we are seeing here is that the conductivity, how water conducts electricity, depends on the charge fragment formation. So that is actually the mechanism by which the water ionic solution conducts electricity. I have done this experiment probably over 200 times. You know, these are some of the general characteristics of the charge zone. So that's what you've seen so far. You know, there's one Na2SO4. That is the NH4-2SO4. We can see in this case here, there are 2 probes here are at the negative end, and 13 probes went up to the positive end. And that is HCl. HCl is the opposite picture. The 2 probes go up to the positive end, and 14 probes go down to the negative end. So that means that there is where the positive zone and negative zone meets in the middle. That meeting point can vary by a great deal. Here we are seeing that that is 13 on the top and 2 here, 2 at the bottom. There's 2 at the top and 13 at the bottom. And for the NaOH, apparently they all stay at the bottom. It took me a while to realize that, you know, all the probes are not registering any charge zone is because all the negative charge zone all sunk to the bottom. So the bottom layer are all negative charge zone. The positive charge zone are floating on top. We can also see from here that, you know, there is, when I reverse the cycle, the charging voltage, you can see the dip here. There's a dip before it goes up again. The VAB is actually another key indicator. Here we can see that there's 2.2 volts here. There's also 2.2 volts. There's 1.8 here. There's also 2.0 around there. That voltage, the VAB, it depends on the electrolyte. It is not dependent on the charging voltage and not dependent on the ion concentration. It's all dependent on the electrolyte, on the salt used. So we are seeing something very suggestive here. So NS42 plus, there's a SO4 is a strong acid, there's a weak base. Whereas that one here, there is a strong base and a weak, weak electrolyte. So it's actually suggesting that the electrolyte strength is probably having something to do with the charge zone formation. So when the stronger the electrolyte is, the bigger the charge zone is. So maybe I'm just suggesting, maybe it is indicating that the electrolyte strength actually depends on the capacity of the ion to form charge zone with the water. Okay, now this is the conventional picture on how the ionic solutions conduct electricity, the migration of the ions, the negative ion migrated to the positive pole, and positive ion migrated to the negative pole. But if we look at that picture, there's no possibility that this picture can conduct electricity. There's no possibility. So what I am proposing is that if we look at the half equation, there is a term here, there's 4x plus, and that expression is suggesting that the x plus produce here, migrating down to here, consuming at this end. There's a stream of x plus producing at the anode, consuming at the cathode. What I am proposing is that the oxidation at the anode actually produces the X plus, and the reduction at the cathode actually produces the OH minus. So the two of them actually this considers the positive charge zone, and this considers the negative charge zone. And the two zones meet in the middle, in the water body that neutralize each other, and that sort of complete the cycle for how electricity how electricity is conducted in the water. That is a picture here. So the charge zone formation and the meet in the middle and neutralization. So I'll stop here. There's a lot more results published in this paper, so I have to cut short a lot here.
Thank you very much, Dr. Yi. Are there any questions?
It's actually a comment in support of your interpretation, which is, of course, interesting. We did an experiment recently with parallel electrodes in water, and the usual expectation is that the current will flow uniformly from one electrode to the other. So we put a pH-sensitive dye in the water, and we found that next to the negative electrode and also the positive electrode, we found regions, not continuously, but discrete regions along the electrode that began to build up. Next to the negative electrode, they were high pH regions, and next to the positive electrode, they were low pH regions. And these regions would grow toward one another, suggesting, first of all, that the current is not flowing uniformly from one electrode to the other electrode, but actually building up charge regions of positive charge or negative charge next to one or the other electrode. And then when they met at the middle, for a long time, they didn't neutralize one another. One zone would grow into the other zone, suggesting that they might be EZ-like zones that that maintain the charge but don't easily neutralize. So this is a very different way of imagining what happens when you put two electrodes into water, that you don't get a continuous flow of current from one electrode to the other. Very much in support of the kinds of interpretation that you're coming up with.
Thank you. I actually think there are free electrons in the negative charge zone. I believe there is. But I can't prove it,
Thank you. What would happen if you adjust the height of your probes in the conductive channel? But I assume the charge, let's call them clouds, migrate from one for the anode to the cathode and vice versa. They probably form channel-like structures.
No, they don't.
They don't.
They form a big blob coming from one end, another big blob coming from another end. They meet in the middle. And then some pressure will build up, and then there will be some disruption, and one of them will go above the other. So they go into that way. So there is actually grow from both ends to the middle.
So you see this if you adjust the height of your probes across the vertical axis?
I don't have the means to do that, but at some points, I actually sort of make some of the probe longer, and I still get the same picture.
Any more questions? No. Okay, where's our next speaker? Well, we seem to be missing. Yes.
Wise man, yes.
Yes. So
that's it.
So George Weissman. George Weissman is going to tell us some more about brown glass. That's good.
Yes. It's very good to be here. I most sincerely thank Professor Pollack. You've done amazing work. And to be in this venue speaking about the Brown's gas is a great honor for me. As I said yesterday, I am a cowboy. When I grew up, I learned to pretty much fix anything I needed to know. I learned as I went to school. I took all the advanced physics and math and all that kind of stuff. Things you don't usually learn on the farm, but I learned how to make things, how to fix things, in order to do that, you have to understand what it is that's actually happening. And I've learned so much even in this venue. For the decades that I've worked, just in the last couple of days, I've learned even more. I'm very grateful. So, in addition to teaching what Brown's gas is, as far as my theory is concerned, I'm also teaching showing the practical applications of the gas. I'm the kind of guy who doesn't just work with theory. I try to make the theory work in real life. So, what is Brown's gas? Generally, the entire mixture of gas evolving from an electrolyzer that is specifically designed not to separate the gases into hydrogen and oxygen. So, the first theory that we came up with is Faraday, you only have hydrogen and oxygen. Unfortunately, we discovered that we were getting too much gas. For the amount of electricity going into the machine, there was far too much gas, about 60% too much gas. So I came up with a theory, okay, well, maybe the gas, when it's separating from water into HHO, the hydrogens and oxygens are not recombining. That would explain the extra gas, but no, that didn't work. We had the gas analyzed, and then that didn't work. Chris Ekman came up with Rydberg clusters, which was not too far off, and Ruggero Santilli came up with a thing called magnecules. But I found what I call electrically expanded water, which actually is some of these lectures are coming into the same sort of a thing. So back in 1996, I had this very small transparent electrolyzer that I developed, and I was actually able to look in the cells and see what was happening, and I saw something that I'd never seen before, and it changed everything. So what's happening is on the cathode side, we're developing the hydrogen. On the anode side, we're developing the oxygen, just like you would normally think. But right in the middle of the cell, a third gas is being formed. And I call this electrically expanded water. So the electrically expanded water is the unique thing about Brown's gas. It's what makes Brown's gas unique from bottled hydrogen and oxygen. The electrically expanded water is a gaseous form of water, as you'll see in a little bit, that is formed from water in these electrolyzers. So what we have for the constituents, in any water-based system, you're going to have some water vapor. We are seeing some monatomic oxygen and monatomic hydrogen. Most of the gas is the diatomic oxygen and hydrogen, but then we get the electrically expanded water. The electrically expanded water is water that has soaked up electrons until it's become a gaseous form of water that is not water vapor or steam. It's a plasma form of water, negatively charged plasma. So Stanley Meyer, who had drove vehicles on a water mixture and stuff, had a similar type of way of describing the gas in his models here. This is the Chris Ekman stuff where they were saying what his idea was that it would separate the bond angles of the water. I haven't tested that myself, but I thought I would throw that up there just to be a complete explanation. Okay, so here we have a wet mixture. So you've got mostly hydrogen on the one side. I don't know if they've got, oh, it's got a laser on it. That's nice. So over here, in some of them, you can see there's a slight amount of monatomic hydrogen and then mostly diatomic. But right here is the unique portion of it. You've got your hydrogen, you've got your oxygen, but here you have the water vapor. And in this particular case, it is mostly water vapor. But when we dry the gas, we end up still having the water vapor. You can't see it very well in this particular thing here, but the water vapor is still there. So you can see them in this one. You can see better the monatomic hydrogen and the monatomic oxygen, but the water vapor is still there, even though it's not vapor anymore. This is a dried gas. So that's where the proof for the electrically expanded water. It's a plasma form of water that is not water vapor. And also interesting, when they do gas analysis, 7% of the gas, the analyzers can't even analyze. They don't even know what it is. But you don't see those sort of things in bottled hydrogen and oxygen. So, and then the chromatographs of the various molecular structures that happen in the gas. So another thing that we discovered, obviously if you have electrons in the gas and you burn it, the electrons are freed. And this is a characteristic that really helps in a lot of different applications. And in this particular case, we're measuring the amount of electricity that can be gathered from the flame as it's burning. So again, the Brown's gas is specifically the hydrogen, oxygen, water vapor, and electrically expanded water made by electrolysis in an electrolyzer, specifically designed not to separate the gases. If you put a membrane in the middle where electrically expanded water, EXW, is formed, then it will not form. So Faraday never saw this. In all the Faraday 100 and some odd years ago, he didn't see these sort of things. So other names of Brown's gas, William Rhodes called it common ducted gas. Some people called it Rhodes gas. He was the first one that made a patent for this single ducted gas. And we generally call it HHO, hydroxy, or Brown's gas. So knowing what Brown's gas is helps us design electrolyzers that make a higher quantity and quality of gas. We want as much of that EXW as possible. And it helps us discover and develop amazing applications for the gas. So now that you have an idea what Brown's gas is, we go into helping the world with the gas. What can it do? We have theory, but I'm not just a theory guy. Let's actually make practical applications. So we found many practical applications, but I'm just one person, and even a cowboy has limits. So I started with onboard electrolyzers because one of my trades is automotive technician, and I was able to increase the efficiency of combustion of vehicles. I also built large electrolyzers to make the gas replace acetylene and other torch fuel gases, and then more recently the tabletop electrolyzers for health application. This is my brother with his Hyzor system, which is on his particular Dodge Dakota truck, it got a 30% increase in mileage. So the onboard electrolyzers can help the vehicles increase their mileage. As everything you see, it cuts harmful emissions to almost zero. It increases the power and performance. Engines last significantly longer because it cleans them out. The carbon buildup isn't there. The combustion is very clean. One of my pet peeves has always been what's called the catalytic converters, because I feel that burning the fuel in the engine is a much better idea than burning it in the exhaust. That's what this type of technology helps do. There's myself with one of our early machines that replaces the torch fuel gas. So I was able to build machines that were half the size and weight of the machines that were available at the time and produce the same volume of gas with half the electrical power. Again, if you understand what is going on, then you can build better machines. So the Brown's gas flame is very laser-like. It's long and laser-like. It's actually an exploding, imploding flame. It explodes first, it has a flame, but then it shrinks. When it shrinks to water, from a gas form to a water form, it shrinks 1,800 times. So it's literally making a hole in the air, a vacuum, and the air pressure itself holds the flame long and thin as the flame is constantly making a vacuum, and so it ends up being long and thin. It's a relatively cool flame. It only radiates at about 274 degrees Fahrenheit or 129 degrees Celsius. And so you can have it right close to things and it will not burn. You can wave your hand through it, it doesn't burn. If you're using it for a torch, you just simply turn it on and adjust the volume of gas you want. It's already a perfect stoichiometric mixture, 2 parts hydrogen to 1 part oxygen. And it uses no atmospheric oxygen. It produces no toxic fumes, no harmful radiation. It's one of the only gases that's allowed down in deep mines when they're doing work down in there. So the exhaust is just pure water. And in ships too, when they're doing a lot of work at the ships. Now here I'm holding the torch tip, which you would never do with an acetylene torch. Usually when I'm doing the demonstrations, I even put a piece of tape around the torch tip so that people can see that the torch tip does not get hot. And the flame, this is a misconception out there, when they think, okay, you can wave your hand through it so it's this temperature and you put it on tungsten, it goes to 5,000 degrees centigrade. The torch and the flame does not. The material does. So here, the flame doesn't boil water and it condenses, and the new water is the exhaust. And the new water is an entirely different subject I can't even get into right now, but it's absolutely astonishing the things that can happen with new water. The brazing is extremely simple, easy to do, and you can adjust quickly just by moving the torch in and out when you're brazing and welding. You can adjust the amount of power that's going into it. Essentially, think of it as an electrical flame. You're actually putting these electrons right into the material that you're welding or soldering or whatever. So that these are types of things that the flame itself can do. So the Brown's gas is very, very useful in this particular application. So other proven uses, they have neutralization in seconds. They can neutralize concentrated radioactive waste. I haven't seen a way to neutralize, using the gas or this technology, ambient radioactive materials, but concentrated materials, you can get a 97% reduction in practically seconds using a technique that Yul Brown discovered and was using. And this has been proven by even government agencies. The creation of semi-precious stones, now we're talking amorphous, not crystalline, but when you cook the various materials, you can actually make rubies and sapphires and emeralds and things like that. And moonstone from feldspar. Feldspar comprises about 30% of the Earth's crust, and you can actually use this gas to make bridges, for example, out of the stone that would never break or wear or anything. The moonstone is stronger than steel, and if it's pure, it actually is transparent as well. So you could actually make transparent bridges, or buildings, or that sort of thing. Ore and mineral. When you cook the ore with the Brown's gas, you can get more metal from the ore than you could before. One particular copper ore that we cooked got 3 times more copper. You cook the same amount of ore, but you cooked the one sample with the Brown's gas, you got 3 times more copper from the copper ore. So there's a huge use there where a person could go back to old mine sites, and all the tailings ponds and everything where they have gone through the processing plant already. You could cook that and actually get 1 or 2 times more metal out than you originally did. And then quartz and glassware manufacturing is another really good use for it. They were already using hydrogen oxygen gases for this type of thing, but this is really easy to do. So, the health applications. This is where I'm concentrating now, and it's absolutely astonishing. When I first got into Brown's gas, I did not believe that it could do what it does. I didn't have any clue that there would be a health application. I was using the gas for combustion, in combustion for fossil fuels enhancement or combustion in a torch. But for some reason, some of my customers, and one in particular I'm thinking of in 1996, actually bubbled the gas through water, and I have no idea why. And then he put that gas, bubbled water, on a melanoma on his forehead. And that melanoma was gone. He kept changing out his bandages. In 3 weeks' time, it was gone. And it just so happens that that has been duplicated here, like that experiment has been done again in Germany recently. So this is, but I didn't believe him. I didn't believe that particular customer when he told me this. But being the open-minded person that I am, not really, I did tell other people about this potential application, health application, and many of my other customers, which I had thousands by then, started bubbling and reporting back to me. So from 1996 to 2005, I gathered hundreds of testimonials of people who were telling me about these amazing health benefits of the gas. And so in 2005, I decided to do it myself. And so, like I say, and then from 2005 to 2007, I saw a big gain in my health. I didn't get sick in those winters. So I thought, okay, that's good. And I made a tabletop version of the machine, and we started the health application. And so one of the things that people did is they started feeding it to plants. And they found that with regular plants in dirt, if they're fed this water, they'll grow 3 times faster in the same, 3 times more in the same amount of time. And if you do it with hydroponics, they grow 10 times more in the same amount of time. And it's very dose dependent in that the more of the hydrogen that's in the water, the faster the plants grow. And it makes perfect sense because most of us, including plants, all water-based life forms, are mostly hydrogen by volume. So it's our major macronutrient, and it's been totally unknown to medical science all this time. So fish is another one. You can see the 2 fish side by side there. They were grown in 2 separate aquariums. One was bubbled with the Brown's gas water, or Brown's gas, and one was bubbled with just ordinary oxygen, like a regular aquarium, and otherwise identical, and the fish grew 3 times faster, or 3 times bigger in the same amount of time. So you could, it's absolutely astonishing. This is when I started breathing, now, up until 2007, in 2007 and until 2016, my customers were saying, okay, we can drink the water, can we inhale the gas? And I was saying, no, this is a combustible gas. I don't want anybody's lungs blown out on the walls. So but I didn't realize until December 2015, when a customer sent me a video of a Korean hydrogen bar where they were using Brown's gas, and people go in, buy a cup, like we buy a cup of coffee, they would go and buy Brown's gas treatment. And I realized, something I should have known, is if you mix the gas with enough air, keep the hydrogen content under 4%, then it's perfectly safe to breathe and inhale. So in March of 2016, I started inhaling. That's what I looked like then. This is what I look like now. So, and that video's online. So my current mission is to help people regain and maintain health using this specially treated and reformed water. As I was saying before, we're mostly hydrogen, 62% by volume, not by weight. Hydrogen's a very light molecule, but by volume. So, in my experience now, people are sick, aging, and dying from dehydration. Literally, not enough hydrogen. Brown's gas actually helps bodies heal. It doesn't heal itself, by itself. It's not a drug. It's a nutrition, it's a food. And what happens is you give the body what it needs, and the body heals itself. So people using Brown's gas heal quickly, seldom get sick, and regain and retain a youthful appearance and vigor. So the traditional health industry has been missing the importance of hydrogen for centuries, and now the hydrogen industry that's coming up is still missing the importance of the electrically expanded water. The electrically expanded water, again, we have a situation where, yes, you're getting the hydrogen, and that's good, but you also need the electrons, the bioavailable energy. People who are sick generally have compromised energy generation systems and or have depleted their energy reserves. So when you give the electrically expanded water, which is a bioavailable form of electrons, with the hydrogen, everything happens quicker. So just a quick medical disclaimer. So here's a partial list of the ailments that have been reported mitigated with hydration, and the items with the stars. And I'm just going to go through these really quickly because I want to leave a bit of time for questions as well. So it's just a... I have quite a story about the libido thing. So what happens as you are, when you're deficient in hydrogen, the first thing the body does, everybody's aware that when you're getting cold, a hypothermia, the body can shut off blood flow to the extremities to preserve core temperature. Our bodies are amazingly designed to preserve life. So when you're not getting enough nutrition of one kind or another, in this case hydrogen, the body shuts off what it considers to be non-immediately life-threatening systems, like your regeneration systems, stem cells, things like that. And then you still don't have hydrogen, it starts shutting off immune systems, so people get sick. And then you still don't have enough hydrogen, well then the organs start to die. And then you die. So when I started inhaling the hydrogen, I'd been drinking the water. Now you could see, from 1996 I was told that there was health benefits, but I didn't start drinking the water until 2005. I didn't start inhaling the gas until March of 2016. So as you can see, I think I'm open-minded, but I'm not really. So in any case, I'm trying to convince people this is a good thing, and part of it is what happened to me. Okay. So when I started inhaling, or when I was drinking, I just simply didn't get sick. But I was still losing my hair, I still was developing neuropathies, I had my warts, my scars, all these kind of things, tinnitus. And when I started inhaling, all of those things started to go away. So I had a heart murmur, and the heart murmur went away, so my organs were healing. I had warts, which are a virus. All my warts went away, so my immune systems kicked back online to something I hadn't had. I'd had the heart murmur and warts since I was a child, and most of my scars as well. I grew up on a cattle ranch. We cut ourselves, broke bones, all these kind of things from time to time. So so I had all of these things from childhood. And yet, as soon as I started inhaling the gas, my heart murmur went away, my heart was stronger, my immune systems got strong enough to get rid of all the warts. I had hand warts and a plantar's wart, they just disappeared. And as far as the organs go, I had neuropathies on my legs and hand, and I have full feeling back in all of that, pain-free. My arthritis is gone, I have full functionality and strength. And this last thing was my scars disappeared, and I found that to be absolutely amazing. It's something that no other technology out there is claiming, is that the scars disappear. And I've not only on myself, but a lot of the other people that are inhaling, the same thing is happening. In fact, one guy who works with us had a motorcycle accident, and most of the skin on his arm had been taken off, and he had terrible scars. And the scars are completely gone, and now even the skin tone is coming back. You can see the whiteness of the skin, new skin, but now even the skin tone is coming back, and I'm thinking by next year you'll never even know he ever had a scar on his arm. As well as that. So but the results will vary. This is my disclaimer. Every body is different. Literally, every body is different. So how it affects you personally is going to be different. This is the machine that I've developed over years of time, and I'm pretty proud of it. It's a good machine, the AquaCure. So the Brown's gas hydrates, as in giving the hydrogen to living bodies better than any other known technology, because it's not just hydrogen, it also has the electrically expanded water. And that's the main thing I'm trying to teach today, is about the electrically expanded water. It's that gaseous form of water. It's actually water, but it's not water vapor or steam. It's soaked up water that's soaked up electricity until it's become a gaseous form. It's a plasma form of water. So, questions? Thank you. Anything can fail, but I try to design my machines. That's one thing about growing up on a ranch. Anything that would break, I would try to figure out why it broke and make it better so it wouldn't break again, because I like to be able to just use tools. I didn't want to have them. So that's the way we designed our machines as well, so they have a lifetime guarantee.
Thank you, George, for a fascinating presentation. So when we do an experiment with two electrodes, I was just describing at the end of the last presentation, what we find is that EZ grows from one electrode and the protons grow from the other. At the center, you have a mixture of those two. So at the risk of sounding like a guy who has only a hammer and everything looks like a nail, I'm wondering whether it's possible that your so-called expanded water is actually a combination of EZ water and protons. When the two are together, the amount of energy is enormous. And when the two combine, they form water. And so I'm wondering whether there's any possibility whatsoever that what you call Brown's gas is actually a combination of negatively charged EZ water plus protons, which when they combine, they release an enormous amount of energy, and that energy could be used for healing. Just wondering.
Yes, I watched that last lecture with fascination as far as the blobs going out and stuff like that. That was one of the things I was learning. And yes, I want to experiment and find out exactly what it is about that. We know the gas exists, we can measure it physically, but we still need to learn more about it. And in Germany here, actually, there's some people who have developed technologies that can actually look at the gas in a gaseous form and as separate from water vapor. As you saw, the spectroanalysis shows it as water, because it is water, but it can't show it with the extra electrons or energies. And now they're developing technologies where we can do that. And fortunately, I'm in a group that of scientists and people who don't throw the baby out with the bathwater. A lot of scientists say if we can't measure it, it doesn't exist. But in our group, we say, okay, we see it exists, now let's figure out how to measure it. Thank you.
Thank you very much for a very beautiful lecture. What if you compress this gas? What happens?
Oh, don't compress the gas. It contains its own energy already, like an electrostatic energy. And if you compress it, it will explode. Okay? Even if, like, you know how electrical charges form on the edges of sharp edges or points? Okay? Static electricity. If you put a very sharp needle in the gas, it will explode the gas, because the electrical energy in the gas will concentrate on the needle and come down. Again, everything looking like a hammer. It's probably EZ zone type technology that's going. So anyway, we use the gas on demand. We make it and use it. We don't compress it. We don't put it in storage containers, unfortunately.
Can it be used as a fuel in this case?
It can be used as a fuel. I have run engines on it. In fact, one of the reasons I got into the Brown's gas originally is because I was trying to figure out a way to make engines run on water. But I haven't yet figured that.
Not inflammation, but compression.
Yes, you can do it. It runs fine in a diesel engine, for example. Yep.
Okay. I wanted to ask you following. You told that chronic infections like Lyme disease can be healed with this therapy.
Yes.
Which proofs do you have that the germs really die?
Okay. First of all, the gas itself doesn't kill any germs.
Yeah.
If anything, it's as healthful for the germs as it is for the people. So because again, the germs are made of the hydrogen as well. What the gas does is increase the body's own immune system. And the immune system then kills the germs. So, and the proofs are, they have over 1,200 scientific studies now of, I think it's over 170 ailments. Am I getting that correctly? Oh, okay, so many more studies than that that I was aware of. And virtually every ailment known to man, but it's the body itself that is healing. And in all of these studies, where they're now, medical science is now catching up with the hydrogen, they find 0 negative side effects, with the exception of if you have a cold, for example, and you have a virus in the lungs, and you're inhaling, the virus likes the gas and can actually become worse. So a person has to, like any therapy, apply it appropriately. So the proofs are, there are several scientific studies, and if you're looking for any particular one, like bacterial infections, then you would go and look in the medical science. There's now thousands, or hundreds, soon to be thousands. The amount of studies have gone up logarithmically, and it's just amazing. All over the world, they're doing it now.
Okay. It would be possible to get some studies from you about what you say.
Studies, I don't provide the studies.
Yeah.
I provide the machine.
Yeah,
and and so if you want to know the studies, then just go online and look up what you need.
So, okay.
Yep.
Is our next speaker here? Yeah. Where is he? Vladimir, are you here? Ah, you want to get mic'd?
Yeah.
Pardon? My age, I'm 61 years old. In just a few days. Yes.
Okay.
It's an honor to meet you, by the way. Thank you.
Yeah. Okay.
Thank you.
So.
Okay.
So a big wow. Do you have any information on the lifespan of the fish? Because some organisms that thrive very fast also die very soon because of their increased metabolic rate. So it sounds like a fountain of youth, but maybe it should be studied systematically on fish or plants.
Yes, good point. As far as the plants and fish and animals so far that we've discovered, they live longer. So not only did they grow faster, and now I have some people ladies who are pregnant actually and inhaling the gas. It's going to be interesting for me to see what happens to the gestation. But chickens, for example, when they were given this water, they doubled their egg production overnight. It was incredible. Now, when they continued getting the water, they went back to the regular egg production. But for a few days, it actually doubled the egg production. So there are things that happen. But as far as the fish and stuff go, they just seem to grow faster, but they only grow to the size that they would normally if they were an adult, and then they live a longer lifespan. The plants, the animals, everybody. And so my studies, for example, using the hydrogen, are showing a 30 to 50% life extension. So it's pretty amazing. Again, it comes back to nutrition. Hydrogen is our most important macronutrient. And we're not doing any magic here. We're just giving the bodies what they need, which they normally don't get in such a concentrated form.
Yeah, okay.
Yes?
I wanted to make a comment. I and someone else tried the Brown's grass inhaling last night.
Yes.
And we tried the machine that had different frequencies. It was made from different frequencies.
Yes.
The first frequency that was tried, the other at 432, the other person really got a very strong beneficial effect almost immediately.
Yes.
And then I tried the 528 first, then went to 432. At 432, I started to get, my throat started to be itchy, and then I got shortness of breath. But at 528, I felt it was very good for me. So the 432 was not good for me. So I did have a negative side effect, but the 528 I thought was very good for me.
Yes.
So I just wanted to point that out.
Yes. So it wasn't the gas that was giving the negative side effect, it was the frequencies. Exactly. And that's one thing that we did, or I did, because I could, is I made the machines so that they could vary the frequency, because even though I don't understand all the health benefits of frequencies, I do know they exist. And then people who do understand, they can adjust the machine, and it does imprint the frequencies on the water, and the frequencies are read by the body, and they do things. And in some of these lectures that I've seen these people, it's absolutely fascinating, all the different things that can happen. So yes, you can, but you have to be careful, because it can go both ways. If you get the wrong frequency, you can be less helpful.
Quick question.
Yes.
Has anyone done a comparison between hydrogen gas and Brown's gas?
Yes. Now, not officially, and I'm gathering that evidence. See, what happens in all these hundreds of studies that have been done, a lot of them were done with pure hydrogen, let's say from bottles, or PEM technology, which separates the hydrogen and oxygen gases. So they were getting the pure hydrogen. But many of them, about 25% so far as I discovered, were actually using Brown's gas. So I'm now sorting through the scientific studies, I'm having people do it for me, and separating out the ones that have already been done using Brown's gas, and then I can compare, like say, a wound or whatever to healing, a Brown's gas study to the straight hydrogen studies, but it has been done, they just didn't know they were doing it. Thank you.
Thank you very much.
Another thing is the banquet tonight will be in here. So we can't have anybody here between 4 o'clock because they're preparing this whole room.
Vysotsky, yes, this one.
Yeah, so you will say that?
Can you make full screen? Yes, a full screen. Oh, maybe. Okay. And where is the... Okay.
Okay. The next speaker is Vladimir Vysotsky, who will say something about the generation of undamped traveling temperature waves and stimulation of controlled nuclear fusion in experiments with water jet cavitation. Yeah.
Dear colleagues, it's very pleasant to be here and present our new results related to such abnormal phenomena. It is a combination of water phenomena together with nuclear phenomena, X-ray generation, so on.
Just a second. 1 second. Sorry, we just have a quick announcement from the organizers.
Just real quick, we would ask everyone after this next session here before lunch to remain in the room here, because we're going to take a group picture here. And I want to make sure that nobody gets cut out of that opportunity. The other thing is that our banquet this evening will be in this room. So between 4 o'clock and 7 o'clock, no one's supposed to be in here because they have to arrange everything for us here. That's all. Thank you.
Thank you. Okay. So, this is short contents of the report. In the report will be discussed, first of all, the problem on non-dissipative distant energy transfer by undampened thermal wave. What is a thermal wave? It is absolutely new phenomena. Also the method of generation of these waves, and the most important may be the distant action of this wave, including influence on the optimization of nuclear fusion in distant target. So, the problem of energy transfer without loss over long distance and the problem of efficiency use of such wave is among one important. As usually in applied nuclear physics, and not only nuclear physics, using electromagnetic wave in the form of generation of giant, very short laser pulse. but creation of such pulse is a very difficult, necessary, very difficult problem, very great efforts, and also it is very expensive problem. Very interested, we have a study, investigated phenomena of that connected with bubble cavitation, and also such phenomena we have during our investigations that were presented in some very old works. We have obtained very anomalous optical and X-ray phenomena that connected with direct motion of running liquid at a relatively low pressure through such system. For example, it is initial our experiments. We have the circulation of machine oil. In first experiments was machine oil pump system and also cavitation chamber, very thin diaphragm or rest hole, and it is an area where the process of cavitation takes place. It is a general view of such cavitation chamber made of plexiglass, and these three photos show it is a photo of cavitation chamber at different pressure. At relatively low pressure, we have the motion of machine oil, similar milk. This oil is situated in such area. And increasing pressure, we have the process of formation of direct stream. But most interesting, that pressure about 80 atmosphere, we have that such machine oil has a very brightly lighting, similar, for example, sunlight. Very interesting phenomenon. Why? For example, the same photos. The important question is the following. What is the nature and origin of this directed luminous beam? It is easy, short analysis shows that it is not usual sonoluminescence, because sonoluminescence is very slow, very weak illumination and absolutely different way. Also, it is not temperature radiation, because the machine oil is cold all the time. The answer to the question, we investigated radiation, first of all, radiation outside of this cavitation system. For registration, we're using the X-ray detector. It is a detector, X-ray detector, that was situated immediately near surface of such cavitation chamber. Is what. And very interesting, in our first experiment, we have observed that the external surface of plexiglass chamber in the cavitation chamber, in the center of which was cavitation phenomena, is a source of X-ray radiation. X-radiation, spectrum X-radiation depends from the pressure. For example, at low pressure, relatively low pressure, it is such peak is some less than 1 kiloelectron volt, and increasing up to 60 atmosphere, the energy about 1.5 kilo electron volt. And much more pressure, the X-ray radiation is absent, but we have observed new phenomena, lighting, machine oil stream, very strange phenomena. Okay, during a long time we studied the nature, reason of such phenomena. And we have using the acoustic method of analysis. For measurement, we use the piezoelectric converter that was situated on the surface. And we have observed that during the process of application, on the surface registration, quasi-periodical short pulse, acoustic pulse, very great intensity. So, the mechanism of X-ray radiation generation was connected with sequential tandem of cavitation, first one, and shock wave phenomena inside the chamber. For example, in this area, it is a process of formation cavitation. Also, cavitation lead, after finish of process of cavitation, formed shock wave. Shock wave traveling through wall. And during interaction of shock wave with atoms on the surface of this cavity, of this plexiglass system, the generation of X-ray take place. It is a typical method. We will know the dynamic or evolution of bubble cavitation from initial process of born, bubble in the compressed liquid, growth of bubble, and also we have the final state. It is a collapse of cavitation bubble, a generation of acoustic wave. It is a scheme of transformation of energy of bubble water cavitation collapse to the outside X radiation and indented thermal wave generation. First one, it is a formation of cavitation. Next, it is a formation of acoustic wave. Next, transformation of usual acoustic waves in shock wave. Next one, interaction of shock wave with internal surface of wall. Also, formation of acoustic and shocking wave inside, and reflection of shocking wave from external surface lead to excitation of atoms and generation of X radiation. Many steps process. And also, what is the reason of formation of X radiation? It is following from the usual law of quantum mechanics. If we have the sudden acceleration of any atoms, so in initial case, we have the initial, for example, wave function of atom that is not moving, and it is a wave function of the same but moving atom with a sudden acceleration. And also, it is easy to calculate the probability of excitation of atom during such acceleration, and it leads to the probability of excitation atom. This result is a very good correlation with our experiments. Okay. Now we have a study way of optimization such radiation. For example, on this case you can see it is a peak of a spectrum of without copper power. It is a cross section of cavitation system. And also, if we to add the powder of copper on the external surface, copper, we have generated additional, much more intensive peak with much more energy, up to 4 kilo electron volts. So of generation of X radiation is external surface. It is a result of many steps process of transformation, shocking wave in elastic wave, and so on. Okay, this experiment we have conducted about 15 years ago on the base of the machine oil. But machine oil, it is not good object for investigation, because during such circulation for a long time, the process of heating of machine oil take place first, and decrease of molecular weight, and decrease and changing of properties of machine oil. So it is bad objects. Now our next step was investigated the similar effect using the water cavitation. In our initial cavitation experiments, we have using the so-named KMT industrial equipment. It is equipment for cutting and drilling of thick up to several centimeter metal and ceramic plates by water jet. There is a water jet. Here is with high pressure from several hundreds up to 2,000 atmosphere. And it is an industrial equipment. And we used such equipment for analysis of possibility of realization the same effect. And it is investigation spectrum in pure X-ray radiation by fast free water jet. It's approximately geometrical system. It is some road with a hole inside, and it is a water jet under different pressure, and immediately near such water jet in different place was situated of windows of X radiation. And next result is very interesting. This is a different place for pressure about 300 atmosphere left and up to 600 atmosphere. You can see different form of the water jet, of course. But first of all, we have observed absolutely anomalous effect. The pure water jet is a source of X-ray radiation. If we have changed the, I repeat once more, if we had put such windows of spectrometer, in, for example, in this place, we have registered X-ray radiation. So external surface of X-ray, of water jet in the state of cavitation is a source of X-ray. What is the reason of such phenomena? Very simple. During process cavitation, the same process takes place. First of all, formation, it is a cavitation, generation of acoustic wave, transformation of usual acoustic wave to shocking wave, traveling shocking wave during the during the diameter in cross-section of water jet up to surface, and reflection of shocking wave from surface of water and excitation of atom on the surface. In result, water jet is a source of fixed radiation, absolutely abnormal phenomenon. Okay, it is a view of the X-ray radiation at, for example, water jet with pressure about 600 atmosphere, and it is a X-ray radiation from the steel surface of the rod, because it is a spectrum of radiation depends from the material, because it is hydrogen and oxygen, and energy X-ray radiation is low, about 1 kilo-electron volt. Because there is a ferrum, iron approximately, so the energy increases up to 2.5 up to 3 kilo-electron volts. Also, if we had on the surface, if the road was covered by fine dispersion plume bomb powder, energy of X radiation increases up to 5 kilo-electron volts, because on the surface will be situated heavy atoms, and action of shocking wave on this atom lead to generation much more intensive and much more energy X-ray quantums. Okay, next step of our investigation, it is, for example, we have shown using not only X-ray spectrometers, but also X-ray films. X-ray films, for example, it is the same industrial equipment, and it is two X-ray films situated without clearance in the cylindrical geometry, for example, such, around the cylindrical rod. And you can see the darkness, very strong darkness, black signal on this upper part of such X fields that's situated in this such upper part. Because this distance is such, this rod has a different cross-section, and in this case distance was low, and we have registered such radiation. There is distance much more, and without such radiation. The structure of X-ray signal fixed was the same geometry, but it is internal X-ray films, it is external. Okay. After we have conducted additional new type of experiments using some modified system similar to this. The cross-section system or scheme system is the following. It is also water under pressure, different pressure, and also cavitation chamber, formation of, it is a closed, in this case it was closed chamber, formation of cavitation bubble, and also interaction of such water stream with cavitation bubble with target. And we have investigated different phenomena behind such target on this area. In these experiments, we have observed absolutely unknown for us, and not only for us, phenomena. It is a generation on very large distance, very high frequency wave. Frequency of this wave was, minimal frequency was about 80 megahertz in air. And such frequency was detected at different distance. For example, it may be 1 centimeter, maybe 5, 10 centimeter, 50 centimeter, and up to several meters. Magnitude of such waves was approximately the same, not decreasing. In fact, it not decreasing. it is very strange phenomena because, well known, yes, of course, what is a method of registration? We used the usual piezo detector that using for detection of ultrasound. But well known that hypersound with frequency about 100 megahertz cannot distribute it in air. Well known that the free pass of such hyperzvuk is several microns. But we have registered such wave on the distance up to several meters. A detailed study has shown that this process are associated with the generation and propagation of previously unknown high-frequency undampened thermal temperature waves. It is very interesting from the standard thermodynamics. On one hand, the solution of the standard equation of thermodynamics lead only to very dampened waves, both in the case of monotonous changing thermal field and the case of thermal waves. In accordance with the thermodynamical canons, such waves are very rapidly dampened, passing into an instrument in very short distance. I repeated once more said that for frequency up to several tens megahertz, up to, for example, 100 megahertz, free pass for free traveling is several microns, but we have observed by up to several meters. On the other hand, it is easy to verify that the standard classical equation of thermodynamics were obtained with rather incorrect assumption. It's error was made about 150 years ago when classics of thermodynamics create classical thermodynamics. The historical mistakes lead to the fact that the probability of realizing undampened temperature or thermal waves, which can propagate without loss over a long distance, was lost, unfortunately. Let's consider very, very briefly the standard way of creation of equation of thermodynamics and more correct. The traditional or standard concept of the laws of thermodynamics is based on assumption that the propagation of thermal excitation is a purely diffuse, incoherent, irreversible nature. Mathematical description of these processes is based on the joint usage of two basic equations. First one, the Fourier law for non-stationary heat flux, this one. And the equation of continuity, second one, where is the parameter, it is the coefficient of heat transfer, it is density and the specific heat. It is traditional calculation. And also, from the two equations, the classical parabolic different equation for a special temporal variable is the temperature field follows such equation, this equation. This equation, it's very easy, any student study on the third course of study, such equation in solution. This equation has standard solution, two colliding plane wave with very great damping coefficient. Damping coefficient is equal to the wave number. And for example, if we have frequency 80 megahertz, this frequency we registered the experiment is following from our next calculation. We have the damping coefficient in 10 power 4 centimeter minus 1, and the very small free pass, about 1 micron, only 1 microns. It is a standard. This equation you can see in any handbook on thermodynamics. In more realistic case, of finite time, non-zero local thermodynamic relaxation, the heat transfer equation with delay has a different view. What's the difference? In the Fourier law, a very not correct assumption take place. So we have connected passing energy in such place and increasing temperature in such place. It is a process take in some moment, but is it not correct? Because the process of formation temperature needs some time of local thermodynamic relaxation. So if we take in attention such fact, we have absolutely new modified equation, additional time of relaxation here in this equation. The solution of this equation is also superposition of two colliding waves, but with absolutely different coefficient of damping, controlled coefficient of damping. It is, for example, this equation different, fundamental different from the solution of standard equation, because with fixed coefficient absorption, it is a controlled coefficient absorption. Its coefficient depends from the frequency of wave and time of relaxation, and also the thermal diffusivity coefficient. So there is specific, some very interesting frequency on which such coefficient equal to 0. Yes, okay. So if the frequency of radiation equal to such expression is proportional to relaxation, and it is a parameter maybe 1, 2, 3, and so on, we have the result that we can register it or generate it wave without any absorption, any damping. For example, such characteristic frequency in air, this about 80, approximately depends from the pressure, 80 gigahertz, in water about 100 gigahertz, in metal about from 10 up to 50 terahertz. And also, we have investigated the process of reflection, scattering, so on. It is not important. Important is the following. First of all, how we can generate such wave? Because frequency is very, very high. Of course, if we can using modulated laser beam with such frequency, it is possible. But it is very difficult. Most optimal wave, it is using cavitation phenomena. Why? Because during cavitation phenomena, we have generated very short pulse of acoustic pulse, shocking wave pulse, and heating pulse. If the duration of pulse is very small, among the Fourier components of such pulse is present the component with necessary frequency. So we have conducted such additional experiment, for example, this experiment, but you can see there is a spectrum of undamped wave on different distance, 80 centimeter peaks, 46 centimeter, 2 meters, up to meters. But distance to meters is limited on the size of the laboratory, because such wave we can observe by distance many, many meters. Very interesting. And also next part, last or final part, is a controlled neutral processes stimulated by the action of undamped wave. One of the possible applications of undamped thermal wave is connection with direct distance stimulation of nuclear processes. It is a scheme of our experiments. It is the same cavitation chamber. It is the same screen. Generation near the surface, generation X-ray radiation, of course, but X-ray radiation is situated on a very small distance, about maybe 1 centimeter on the, and also we have generation undamped thermal wave. And we using the distant titanium deuterite target, loaded by 150%, and also it is two track detectors that situated. Distance was different, it will be from 10 centimeter, 20 centimeter, and more. And for carry out, we using alpha track analyze a plastic detector with such systems, also on different results. And also on this figure, these photos, you can the result from track detectors. You can see that detector, once more repeat please, one detector, it is target was made in cylinder, was plastic detector was situated near such surface, and another one near cylindrical surface, both. And on this figure you can see track detector, tracks, alpha tracks. This plastic detector was situated on the near the non-cylindrical path. You can see that direction of motion of tracks, direction tracks, has a cylindrical geometry. It is a near cylindrical track situated near cylindrical surface. It is a dark, so there is no directed motion of alpha particles. And this figure, it is a control photos made the control sources of radionuclides, uranium, plutonium, and we have absolutely the same tracks on both systems. It is the same very interesting tracks. You can axial symmetry of tracks under the action of a shocking wave. And very interesting, what is the physical reason? Because you know, now the problem of controlling nuclear fusion, it is a very difficult problem. Necessary special tokamaks, the cost of creation of such tokamaks is $10 billion, but we have the same system at infected room temperature. It is the following interesting fact. In fact, is connected with the following. During the process of loading deuterium in the target, in the titanium target, the microtracks take place. During action of shock of distant thermal wave, the process of formation of local shocking wave takes place. The shocking wave leads to modulation of size of such microtracks, and it leads to formation so-named coherent correlated states. It is very interesting, coherent correlated states. It's absolutely new states. In such states, the process of synchronization of different fluctuations take place. So it is easy calculated showed that fluctuation of energy, if we have usual system, deuterium situated in any microcrack, the fluctuation of energy is on the level 0.1 electron volts. But if the formation such action, the fluctuation of energy may be up to 100 kilo electron volts. On this figure, you can see the method, ideology of formation of such correlated track. It is uncorrelated track. If we have particles that situated in a potential hole in superposition state, the state of different aging function is totally non-correlated. It leads to the fluctuation of pulse and energy is very small. If we have formation of correlated states, we have generated giant fluctuation of energy. Fluctuation of energy may be increasing many hundred kilo electrons volt. Very simple analysis. All man consists and our body consists from molecules. Molecules are in moving state. Velocity of moving about several hundred meters. My question, why we don't fly because such velocity? Answer very simple. Because the motion of molecules is totally stochastic, random. If we can organize correlated, synchronized motion, we immediately will be similar plane. But it is impossible for macroscopic system. But the microscopic system, using the quantum mechanics law, it is possible to make such synchronization and generation giant fluctuation of energy. Such fluctuation energy is a source, for example, by using the quantum mechanical calculation, using the increasing transparency that, for example, at usual case for room temperature is only 10 power minus several thousand. It is approximately zero. It is impossible at room temperature create fusion. But in the case of correlated states, it is possible the coefficient of transparency will be close to 100% at any temperature. It is absolutely new physics. And also, there is a very long list of articles that describe such possible of the mechanism of creation of correlated states, formation correlated states, a different modulation. For example, it may be periodical modulation, increasing, decreasing of surface action, pulse magnetic field action of shocking wave, so on. And also, I have only 2 slides, yes? Okay, yes. And also, it is a this is an experiment that we conducted immediately during the last year. It is a temperature wave, X-ray and controlled fusion behind the remote steel screen. We have additional possibility. What is the difference? In our preview experiment, we have the same chamber and was created in approximately this place. It's titanium deuterium target. But in this case, we have used additional metal screen, investigated fusion target behind the metal screen. Behind. It is very interesting, and we really it is a result of registration of distant thermal wave. And also, on this figure, you can see signal on track detectors that situated near the titanium deuterium target that was situated behind the distant screen. You can see cavitation chamber, screen, generation of thermal wave, metal screen, and behind the screen there is titanium dead target. And we can simulate nuclear fusion in this distance screen. Very interesting phenomenon. And also such conclusion, very, very long 2-page. First of all, it is description, what is the precondition for generation of X radiation. Very interesting, because we can create X-ray generation that might be used, for example, in medical application, in stomatology, because in this case, not necessarily using high voltage, so on, or radioactive isotopes. No, it is only pure water jet in the closed chamber. And also it is a description of what is the precondition for simulation of nuclear fusion in the distant system. Okay. So, thank you for your attention.
Well, thank you very much for...
Unfortunately, I have no time for more detail.
Thanks very much for a fascinating talk. Are there any questions?
Thank you. Very interesting report. We used water jet, but what can you say about acoustic cavitation? Will the picture the same or no?
The answer is the following. If you have the acoustic cavitation, it is necessary mechanism, is possible, but the effectiveness will be less. Why? Because in this case, for most important, creation of shocking wave with very thin front. In condensed matter, for example, in water, the thickness of front of shocking wave in maybe 1 nanometers. In air, it is absolutely impossible. It is will be very long shocking front, and so will be much more weak action. But effect will be the same, but will be less by several order, I think.
Thank you very much. A remarkable presentation, and I can thoroughly recommend all of the papers that you listed there. I have read through them all pretty well. I'm struck by the similarity between the undamped temperature waves that you observe and the strange radiation that has been observed by many people, particularly working in Russia. Could they be related in any way?
It is not questioned. It is an objective result. Not another wave among electromagnetic, of course, but it's not electromagnetic, that can traveling for long distance with frequency 80 megahertz is minimal frequency. Now, in fact, there is very discrete spectrum, and maximal frequency that we have observed 2000 megahertz. It depends only on the duration of pulse. If we have short pulse, acoustic, short shocking wave pulse, in spectrum of such pulse will present frequency up to very high frequency. So in this experiment, we have resisted thermal wave with frequency 2000 megahertz. It's absolutely impossible for any acoustic system. It is not hyperzvuk, it is hyper sound. It is a fantastic frequency, yes. But we have observed now for a large distance, 2 meters. Meter is the fantastic distance for hyper sound, because hyper sound usually moving on several microns.
My question was exactly about the exact experimental method to measure the temperature waves. What is the exact experimental method?
Yes, of course, very interesting. Of course, we have not thermometer or subsystem for measure temperature with such very with possibility change such very sharp increasing of temperature. But action of temporal wave, if temporal waves can distribute it without absorption, for example, in air at such frequency. On the body between air and, for example, internal windows of X-ray radiation, the process of absorption, such wave take place, absorption and reflection. It leads to periodical deformation of windows. Its windows registered as acoustic waves. So we have using the acoustic detector for registration of high frequency temperature waves. Because not absent system with thermistor with such high good resolution in time is absent. It's a question for future, of course. We're using piezoelectric detector for our experiments. It's very good for our analysis. Very sensitive, we get a very good solution, a very nice resolution.
Thank you for your presentation. Just a comment, maybe. I am pretty doubtful about the fact that these waves are thermal waves, because if the propagation velocity is independent by the mean, it means that the carrier are not phonons. And I think that I read some years ago a paper about the production of unknown kind of waves by the very strong spikes made in superconductors that were able to move masses, pendulum, at certain distance, also tenths of meters. They call the scalar waves, but I'm not pretending to tell that they are. But I think that in these shocking events that you are creating, yes, maybe you are dealing with this kind of stuff that maybe we don't know yet well, to which are also associated maybe some thermal and acoustic phenomena. Just a hint that I want to do. Tell me what you think. Maybe it's wrong, but...
Yes, first of all, I repeat once more, it is not acoustic phenomena, because it is absolutely impossible generation and propagation hyperzvuk with such frequency, several hundreds megahertz, impossible. Related to the velocity, on these slides, one of the slides was formula for the phase velocity of such wave. It depends from frequency. For example, at frequency about 80 megahertz, velocity is very low, about 40 meters per second. It is a very slowly wave. In increasing of frequency, velocity will be increasing also. And also, we have registered phenomena. We're using several separated detectors that situated on several distance from source, and registered the process of delay from one register system from another one. And so by this difference, it easily calculates the velocity. It's very good correlated with our analysis of velocity.
Okay. With that, thank you very much for your interesting. And with that, before we go to lunch, let's all gather for the picture, if I understand.
Couple of more announcements, please. So first, tomorrow dinner will not be set up as a buffet because not many people are staying.
Hello, dear audience. We have the unfavored situation that we have to start because we have to leave at 4 o'clock this room. So we cannot press it far further down. Everybody has now around 6 minutes to present their company and their product. It will be a kind of rush. We have all the sponsors right here, so we will make not a long introduction. Everybody will introduce themselves, and in the 6 minutes, we will try to bring all this information over about why we are here, what we are doing, and so on.
Yes.
No, it's
Hi, everyone. Can you hear me? Yeah? Great. So my name's Meher Engineer, and I'm here with my mother, Thriti Engineer, who's the founder of the Super Coherent system. And we are here with the Super Coherence water frequency, which is this little chap here. So this is going to be very quick. So if you'd like to have any more information, please do come and visit us. We're just opposite the restaurant entrance. So obviously, big thanks to Professor Pollack and all of the people who've organized it. So I'm going to be really quick, just like the supercoherence water frequency, which is very super simple, very quick, and it's also very easy. So there's a lovely quote, which is a paraphrase of what Einstein said, which is, everything should be made as simple as possible, but not simpler. And that's Roger Sessions paraphrasing Einstein. So I think all of us here recognize that there's an urgent need for practical solutions to help heal our water, which is basically in crisis on our planet. I don't think there's anybody here who would dispute that. The water frequency is unique in many ways. There are many ways to restructure water, and whatever you choose to do, you know, I think we're all aware that it's important to do that for our water to make it more suitable to drink. So this frequency is, what can a piece of glass do? What is it? It's glass, but it's got information. And that information is very precise. It's very strongly held, so it keeps its informational integrity. In heat, with X-rays, any number of people can hold it. It's very simple to use. It can be put in the water. It can be put underneath the water. It's very quick. Our studies have shown that it restructures water anything from 2 to 5 minutes with a jug. For a whole house, it takes about 4 days. And you just have to tape it to the mains pipe of the house to do that. So the other thing is that it's very portable and very versatile. And one of the things that makes this really unique is it's the latest addition to our family of supercoherence frequencies. And it is evolved from a set of frequencies that we call the return to love frequencies. So everybody yesterday who talked about that four-letter word love, we are very much with you on that. So we say it's good for you and good for the planet. So somebody asked me, why is it good for the planet? And the reasons are, there are no negative trade-offs. It's in glass, which is inert and non-toxic. It lasts, as far as we are aware, a lifetime. The water that goes down your drain is structured with this, and we know that the water that's structured with this, when it's put into bulk water, will restructure the bulk water. So it's a sort of knock-on cumulative effect. So what are our key messages? Our key messages are that clearly the water that we have is under extreme stress, and that has implications for the well-being of all life on our beautiful planet. The beautiful thing about water is that because it demonstrates this high degree of anisodesmicity, it means that it is very susceptible to being restructured very quickly, very effectively, and very efficiently with the correct information. So it's very forgiving, and it's very quick to restore itself with the right help to its original pristine beauty. So I think the beauty of this forum is that there's cooperation, collaboration, and building strong community, which we believe are key to helping heal the water on our planet. And I thank you all for your attention, and we are delighted to be here. Thank you.
Okay, a very quick change. My name is Elke Freyer, that means free, more free actually. And I'm working as a therapist with water, and you know the stall outside, you can test the water. I don't tell you much about the water because I've already talked so much about the inner values of these living waters. The company is St. Leonhard's, and it's a German-based company. Actually, we are south of Germany, in between Munich and the Alps. And the water comes from the Alps. And, well, why is it living water? It's living water because it comes to the surface by itself. And we don't treat the water. It contains natural information. And this is really crucial to us. We think that everything which is done with the water destroys the natural beauty, destroys the natural, what inherits the water, you know what I mean. And we've done a lot of studies for the last 25 years into the depths of the water, especially, I mean, there's the scientific side and there is the consciousness side, the awareness side. And we are trying to know more about the scientific side of our waters. We haven't found much. Peter Darch, who talked yesterday, he's the first one who did a study on the scientific side, how the water affects the cells. That we can present to the science, to everyone. The awareness side is hard to explain to people, and we are not allowed to do that because we are food, and you are not allowed to give any. Well, food has no effect on health. That's what they say. You are not allowed to say anything, or food shouldn't have an effect. But it has a therapeutic effect, the water we have. We've tested them all, you can test it outside. Why, I was asked, why are you here and why do you sponsor this conference? It's an international conference and we don't sell internationally. We only distribute in Germany, Austria, and Switzerland. It's not a commercial reason why we are here. The reason is, I think we belong here because we are, for one, market leaders on the natural food sector. We believe in consciousness, in the consciousness of water. We've done a lot of studies, as I said, and when I came here, I really felt at home because it is like family somehow, even though we haven't been here before. So many people, scientists who are so humble, know a lot but are humble in what they say, and there's a lot of consciousness as well. And water and consciousness, that comes together, and it belongs to the waters we distribute, and we believe in the spiritual effect of our waters as well. Yeah, that's about it. That's why we are here, and that's why I tried to convince the family who owns the company to sponsor the next Congress as well. Hopefully, if you like the water. It's important that it's in glass bottles, I think. And so hopefully we see each other next year as well.
Good afternoon. My name is Robert Pasco, and I represent Crystal Blue Global. We are a structured water unit producing company. I've been coming here for many years, and I just want to start with a bit of gratitude and say thank you. Thank you all for coming here to watch this and partake in this wonderful event. We have listened to the science, watched nature, and are trying to proceed in a way of biomimicry. In other words, we copy nature to produce structured water. In doing so, we use a design with crystal balls inside of a tube that when the water flows through it, it creates a vortex, and it spins the water in different directions. So from that, we decided that we needed to have frequency. That vortex, in our opinion, releases the memory of the water. So then we decided we needed to imbue into the water the proper memory and the proper frequencies. So we calculated and formulated a mineral mix from different types of minerals and gemstones, shungite, tourmaline, and a number of other ones, iridium. We then packaged around the balls the mineral mix, and we used 100% copper inside so that it was conductive, and then we put stainless steel, medical grade stainless steel on the outside so the radiation wouldn't go out but in. So we then decided, rather than be in the laboratory to start with, we'd go out into the field. So we started on farms in India and in North America, proceeded through Malaysia, China, Philippines, many places. And what we found was we got consistent results of 20 plus, up to 100% increase in produce. We got increased nutrient values in the plants measured by refractometers on the brick scale. We got healthier plants. We used less water. So that convinced us, and then we went to the laboratories and we started doing nuclear magnetic resonance tests, which again confirmed what we knew already. So we have found the benefits of it, and basically what we've tried to do is put ourselves in between science and practicality, because the science is there. We know that. We all know that. But if you can't get it to the people, then what is it? It's a great idea. But when you get it to the people and they start using it and it's beneficial, then we have something. So our game plan, or our goal, is to dechemicalize the food chain for starters. And that can be achieved by simply changing the water. We have no more need for pesticides. We can use natural fertilizers. There's no such need for herbicides. And so we have, over the last, say, 8 years, over and over and over again, proved this viable and workable in every region in the world. That's opened the door for us into industry, into the health industry, where we find that, when you're using frequency technologies or energy medicine, water, of course, is vital. The cell is surrounded with water. So that's basically what we've done, and we're ongoing, and we look for support from any way we can. So in a nutshell, that's it for Crystal Blue. Thank you.
Welcome. It's a pleasure to be up here. And this is a sponsored presentation. I'm not exactly sure what that means, except I'm to let you know what we're about, as far as that is concerned. I'm not sure if we were asking for sponsors or if we're supposed to. Anyway, doesn't. Anyone who wants to participate in our technologies, just get a hold of us, and we'll find some sort of common ground, and we can go from there. It works out to, this Brown's gas seems to have applications in virtually every problem area that is out there. I personally started my own company called Eagle Research, and I developed the Brown's gas with several different applications. And we are also having a German company that is now developed, and there's a company in Australia and the United Kingdom as well that are being developed to promote the gas and the applications for the gas. So, and I'm trying to promote as many businesses to start as possible, because the more that are out there, the faster these technologies can change the world. And we talk about nuclear industry, for example, and one of the problems with nuclear industry is the concentrated radioactive waste. And they've proven that you can neutralize the radioactive waste with the Brown's gas in one particular protocol in virtually seconds. So it takes very little electricity, and at a nuclear reactor, they already have, because they're creating electricity, that's why they're there, and they already have water, and that's the only two things you need in addition to the Brown's gas machine. So there's no need to transport nuclear waste anywhere or store it anywhere. It can all just be neutralized. Unfortunately, the problem seems to be political and not like the science is easily done, but the people who own the nuclear waste don't want to neutralize it. And they don't no one tells you this. They want to, it still has energy in it, and they're thinking of making things like nuclear batteries and things like that. So that seems to be the problem in that particular case. We could, with Brown's gas, for example, start just pie in the sky, just thinking about things. We could cut in half carbon fuel consumption in 1 year. Entire worldwide, this could be done in 1 year. Every single vehicle and furnace out there could be modified with the Brown's gas so that it would reduce the amount of fossil fuel that it's burning at the same time as maintaining the heat and power and efficiency. So now some fuels, the heavier hydrocarbons like the crude that they're using in ships, they have shown to be, you could reduce it by 90%. 90%. The ships could be using only 10% of the fuel and still have all the same power that they had before. It's already been proven. So, and the lighter hydrocarbons like methane, we can only get a 5 or 10% at the most efficiency. But so the heavier the hydrocarbon fuel, the more it gains in its efficiency. Then we get the health benefits. If you have the just to take a quick look at the timing here. Okay, we're doing good. If we have with the health benefits, we could reduce the load on the medical systems by at least half. Like myself, I visit the doctor once a year just for a checkup. I don't need to go to a doctor for any other reason. And doctors can mostly become people who fix broken bones and what have you. Most of the diseases that are out there, in my experience and research so far, are either caused by or exacerbated by lack of hydrogen. So if we just simply use the Brown's gas or hydroxy, HHO, whichever you want to call it, to in every home, just like everybody has a car, everybody has a toaster, everybody could be inhaling and drinking the water, inhaling the gas and drinking the water, and all of a sudden medical bills would just drop significantly, and people would have longer, healthier lifespans. Productivity would rise. It boggles the mind all the different things that can happen with this gas and the various applications of it. So I don't. That's me and the technology that we're talking about. Are there any questions? Why don't we go with that? Yes. Okay, so the protocol to transmute the radioactive waste. Oh, by the way, yeah, transmutation is a whole other story that could be happening with this gas. But what you do is you take equal parts of the concentrated nuclear waste and iron and aluminum, and you mix them all together in a crucible, heating them in a puddle with the Brown's gas, which adds the energy necessary for this reaction to happen. And when the puddle reaches a certain temperature, the iron and aluminum form thermite, and there's a pop, and sparks kind of fly everywhere, and the radioactivity is gone. And so what's happening is most radioactivity is caused because they're stripping the electrons off the outer shell of the material. And so when you first of all add electrons back, there's no more radioactivity. It's balanced. But also, the transmutation actually happens. That was a good word, because you end up with a different material. I actually don't know what that material is right now yet, than it was before, but now it's balanced. All the constituents of the atom are balanced so that it doesn't have to radiate gamma, beta, or particles. That was a good question. Any other? Yes? I guess that was the end of my presentation. Thank you.
Hello, everybody. I want to thank the organizers of this meeting. And it's the first time for Salvital, that is the company that I'm representing today here in this conference. And we are not, some of you come to our booth and ask, this is something that is related with water, and in your machine there is water inside. The answer is no. What is doing, what is the high knowledge of Salvital have is about energy and how to deliver this energy in form of frequencies and electromagnetic fields to the body. So what we do is, and this is scientifically demonstrated, is to move ions, especially cations that are solved, that are inside in the water, in the matrix, surrounding the cells, and to activate this to put it inside of the cells. So this is a technology that promotes a lot of processes inside of the cells and, of course, in organs and organisms. And it's something that it's more easy to demonstrate, to make them try it. And I invite you to come to our booth, and I will be more than happy to give you more details regarding how this is working. And for example, one of the things that is related with this to fighting inflammation, and we all know that is one of the basis of what are chronic disease actually in the mother worm. So again, thank you for the organizers, and thank you again from Salvital Company.
Okay, hello guys, it's a pleasure. So we are coming from Blue Boson Company, and our company is focusing on water treatment we are using special kind of frequencies. And thanks to these frequencies, we are available to change structure of the water or any other liquid. And it finally means that we are increasing the heat capacity of the water. We are also able to clean water without using any chemicals. That means we are reducing the chemicals. Also, very often are the pipes or any other sources of water full of incrustation or irons or any other things, and we are available to clean it very efficiently. So that's maybe I will talk very shortly about our vision, because one side is a business, but second side is environment ecology, and we are able to with one shot hit two targets, because as first we do business, and the second it's one circuit. What means circuit about the environment? If we clean the water, it goes through the companies, through the planet, through the humans, through the trees, through the animals. So we are step by step helping the planet. And the reason why we are here is that we know what's the effects of our technology. We have a knowledge how to measurement, but we are looking for more support from you scientists, and about knowing more how it is possible to do in other ways. So for now, I would like to have more discussion. So if you have any questions, please ask us.
You have on your table the advertisement, one page about our technology, how it's looking like. And important is that we have some effects after 100 of installations. And we probably know how it's works, but we want to adjust it and make it more effective. And we are hoping that you know a lot about the water. We're doing some application. And I think that science can help to these practical solutions to bring into the daily life. We need scientists to explain to the people, to explain politicians. Then we don't need chemicals anymore to treat our waters to make it drinkable. We can change the water in processed water in industry without chemicals. And this clean water can go to the waste treatment device, can go this clean water to the rivers, to the sea, and clean rain can fall upon us again. Today we have the problems with emission, but we can change this emission for 20–30% with our device. For even 90% if this brown gas, we have technologies. Why we don't using them? Yes, please. This is purification, like cleaning water. This is not precise word. Like you know, the water is the cluster of a lot of molecules, and we rearrange the molecules. We don't know how it's happened, but it's not. When we use our device, it's not incrustation anymore. We de-incrust existing, and we doing some kind of prophylactation in the water. Yes, this is one of the effects. The same effects is with mangan, with iron, with different kind of particles. We are interested how you perceive our technology and our effort to do something, not because I understand that in business through the science, it's this kind of perspective, it's sometimes not trustable. But if you can see that business can be environmentally orientated, can bring a real change, then maybe you can support us. You have our emails, our contacts in these papers on your table. If you will contact us, we can support you with sources to make your research help us and help, I think, everybody around. Money is involved in this, like in every part of our life. But this is the way how we can help, how we know that we can do some real difference. Thank you.
Thank you. And now the last one, that's me. I'm Hans Eng from the company Eng3 Corporation. We got founded 10 years ago in Seattle, and we are producing the nanobead technology. We are supporting this conference because we depend on you. We are not water scientists in the beginning. We are in the biology, in the medical biology background, and in this area we are especially in the structure biology. What is structure biology? That is an area they have basically two topics. That is a DNA, that is a forming of amino acid chains, and then when these chains are folding, they're creating the proteins. Proteins are the workhorses in the cells for all living species. Without proteins, there are no biochemical reaction initiated, stopped, sensed, terminated, or anything like this. When a protein is not working, we have a kind of biochemical problem somewhere. We are coming from the medical background, so what we produce now is a medical device. It's a registered medical device not only in the US, it is in Europe, it's worldwide used since 10 years. And we at first the idea was to assist the protein folding process. So with our background, we know that the protein folding process is initiated by its environment, and the proteins are in the water of the cell. So the water in the cells are triggering the protein folding process. And then life can begin, life can start. Unfortunately, certain environments of the proteins are damaging their three-dimensional shape, and they have to be refolded. Damaging events are environmental events, radiation, but the most damaging effect happens within the cells, that is a free radical. These guys, when they're going in connection, for example, with antioxidants, and this reaction happens very close to a protein, then it has a damaging effect on the three-dimensional structure, protein stops to work. And we are absolutely not interested to reduce the free radicals, because they are an unavoidable byproduct of the ATP production. ATP is necessary for all biochemical reactions in the body too, so we cannot cut it down. But we are interested in assisting the refolding process, that these guys are getting repaired again, regenerated again, we could say. So many, many years, 20, 40 years, literature is out that describes that the order of the water that is around proteins is actually the trigger for the protein folding process. A thermodynamic entropy change happens when enough of the ordered water is around the protein, and the unordered protein starts to fold. It orders its phase, and the surrounding water is losing this order. So now something has to build up again as a easy water zone, the ordered water around the protein. And we approached this before we were actually in the water science area, this thing by looking what could actually be the source. And we found certain kind of electromagnetic energies are constantly being emitted within the cell. So deep inside, there is no sunshine or anything like this. That is deep in the cell, is always electromagnetic energy emitted. Who is the emitter? There's one specific free radical that is an excited oxygen atom, and when it loses its excitation stage, it emits energy. electromagnetic energy. So oxygen is known, the molecules are pretty much good, well researched, and we tried the first approaches with technology, and we did in clinical studies. We went double-blind, placebo-controlled studies with universities and wanted to see the effect, and we saw tremendous effect with our approach. And our approach was to emit 1200 nanometer electromagnetic energy to vapor and then have this inhaled by people. We said, oh, great. We didn't understand the mechanism. That was why we, years ago, then approached your scientific community for explanations. We wanted to know, how can we keep this under control? Are we doing the right thing? And we learned since then a lot of things. And we did a lot of studies together with different universities. You saw the poster presentation from Olga, and it's still out there. Roberto did studies on other areas, and we learned how to scale, and that we have now other electromagnetic wavelengths that are even more potent. And we can say today what we emit out of the device, the vapor that we treated with this electromagnetic energy, is in a coherent phase. And when this coherent phase hits on a body of water, that could be a petri dish with cell cultures in there, or a petri dish only with proteins, or a human body, the effect, what we would like to achieve, occurs. We are affecting the protein folding process. We can measure those things with different technologies today. And at the big field, we see that the human health is getting improved. So the application of this technology now is used in a lot of areas where regeneration is a focus. Not to avoid a problem, where it is the focus to repair and to regenerate faster. That is not only part in the sport, in the high-end sport, so we have national teams and Olympic teams that are using it, because they can actually recognize it. We have an improvement in restoring the protein function about 20%. That means an athlete is recovered instead of 5 days after 4 days. That is really measurable. The other areas, like in the medical field, to prevent chronic diseases or age-related diseases, which are all caused by proteins that are not executing their function anymore. So it is a perfect prevention tool that is used in a lot of areas. We see that companies are taking in it for their corporate wellness and say, if we treat the people in the company, the treatment time is very short, could be 20 minutes, 30 minutes, 15 minutes, whatever you have time. Then we keep the people healthy. Many, many people recognize the improvement on the first treatment. Some number of people cannot use a treatment for 15 minutes. They can use it only for 2 minutes because the effect is too strong. They can recognize it. And a lot of diagnosis technologies in the health sector are able to measure protein function, and they can all be used. You can see that the blood oxygen saturation is going up while the people are using it. A very good other thing is a heart rate variability that measures this, and a lot of functions are able to be reinstalled. So thank you very much. If you would like to learn more, go to our website. I look for any kind of cooperation with the intent from our side to keep our process under control and maybe to improve it even furthermore for the benefit of everybody in our world. Thank you very much. And I was the last speaker. One thing that I should remind, the organizer asked me, don't forget anything on the tables. They will take the tables out. They have to rearrange the room, and the rest is have a beautiful afternoon. We see us for the dinner. Bye.